Hello there. I'm so glad you found your way here tonight. Right now, as you settle in, something extraordinary is happening within you. Light is being transformed into color. Vibrations in the air are becoming sound. Molecules floating invisibly around you are revealing the scent of everything nearby. Your skin is sensing pressure, warmth, and the gentle weight of the blankets around you. These are your senses, the quiet translators that turn the raw physics of the universe into the lived experience of being human. Without them, the world would be silent, dark, and utterly unreachable. Tonight, we journey into the remarkable world of perception. We will explore over 100 soothing facts about the senses, from the way your brain fills in what your eyes cannot see to the ancient instincts encoded in every heartbeat, from creatures that taste with their feet to the strange phenomenon of hearing colors. There is no test at the end, no rush, no urgency, just the gentle unfolding of how your body speaks with the universe to carry you softly towards sleep. So let your breathing slow, let your thoughts grow quiet, and allow yourself to drift into the peaceful wonder of perception itself. Welcome to Drowsy Discovery. I am glad you found your way here tonight. Perhaps you have had a long day. Perhaps you simply enjoy falling asleep to thoughts about the remarkable processes happening within your own body. Either way, this space is for you. There is something deeply calming about contemplating the senses. Every flicker of light that becomes vision, every whisper of sound that reaches your ears, every gentle touch against your skin carries within it a story millions of years in the making. You are connected to this story more than you might realize. The same mechanisms that allowed your ancestors to sense danger in the dark are still working within you tonight. The wisdom of perception has been passed down through countless generations, written into the very architecture of your nervous system. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is relax. So settle in, let the weight of the day drift away, and let us begin with the first of many quiet truths about the senses. The senses are patient beyond measure. They work continuously, moment by moment, translating the chaos of the physical world into something meaningful. They filter out what does not matter and amplify what does. And yet, within all that constant labor, there is an ancient elegance, a system perfected across billions of years of evolution, a biology so intricate that scientists are only now beginning to understand its depths. Tonight, we will wander through this magnificent sensory world. We will meet the mechanisms that create color, sound, and touch. We will discover how illusions reveal the hidden workings of the brain, and we will uncover truths about perception that may surprise even the most curious minds. But there is no hurry. Your senses have been working since before you were born. They can continue their quiet labor while you rest. Let us begin with what makes perception so extraordinary. Your brain can alter the taste of food based on what you see. The color, shape, and presentation of food profoundly influence how your brain interprets its flavor. In experiments where researchers added tasteless food coloring to white wine, even experienced wine tasters described it using the vocabulary typically reserved for red wine. The brain does not simply receive taste information from the tongue and report it faithfully. It blends that information with visual cues, expectations, and past experience to construct a unified perception of flavor. If a strawberry-flavored drink is dyed green, many people report tasting lime or apple instead. The packaging, the plate, even the lighting of a room can shift the way a meal is experienced. This is not a flaw in human perception. It is a feature, a system designed to integrate as many sources of information as possible when evaluating whether something is safe and nutritious to eat. Your visual system and your gustatory system are not separate departments working in isolation. They are deeply intertwined, collaborating in ways you rarely notice. Every meal you eat is a quiet collaboration between your eyes and your tongue, orchestrated by a brain that trusts sight as much as taste. Some people genuinely taste words or see colors when they hear music. This phenomenon is called synesthesia, and it is not a metaphor or a figure of speech. For those who experience it, sensory pathways that are normally separate become linked, so that stimulation of one sense automatically triggers a perception in another. A person with lexical gustatory synesthesia might taste cinnamon every time they hear a particular word. Someone with chromesthesia might see a wash of deep blue whenever a certain chord is played on a piano. These experiences are consistent and involuntary. The same word will always produce the same taste, and the same note will always produce the same color. Brain imaging studies have shown that synesthetes genuinely activate the brain regions associated with the secondary sense, confirming that these experiences are real and not imagined. Synesthesia is thought to affect a small percentage of the population, though estimates vary because many people with mild forms do not realize their experience is unusual. It tends to run in families, suggesting a genetic component. Far from being a disorder, synesthesia is often associated with enhanced memory and creativity. It is a quiet reminder that the boundaries between the senses are not as rigid as they seem. The sense of smell is more directly linked to memory than any other sense. Of all the senses, smell has the most direct pathway to the regions of the brain responsible for emotion and memory. When you inhale a scent, the odor molecules bind to receptors in your nasal cavity, and the signals travel along the olfactory nerve to the olfactory bulb, which sits just beneath the frontal lobe. From there, the information is relayed almost immediately to the amygdala, which processes emotion, and the hippocampus, which is central to forming and retrieving memories. No other sense has such a short and direct route to these structures. This is why a particular fragrance can instantly transport you back to a specific moment in your past with a vividness that sight or sound rarely achieves. The smell of rain on warm earth, a particular perfume, or the scent of a childhood kitchen can unlock memories that seem to carry their original emotions intact. This phenomenon is sometimes called the Proust effect, after the French writer who described a flood of childhood memories triggered by the scent of a Madeleine cake dipped in tea. It is one of the most beautiful quirks of human perception, a quiet bridge between the present and the past, carried on nothing more than a breath. Every human nose creates a unique scent signature, like a fingerprint. Just as no two fingerprints are identical, no two people smell exactly the same. Your body odor is the product of a complex interaction between your genetics, your diet, the bacteria living on your skin, and your overall health. The genes of the major histocompatibility complex, which play a key role in your immune system, are thought to have a particularly strong influence on your personal scent. Because these genes are highly variable between individuals, the resulting body odor is effectively unique. Studies have shown that trained dogs and even human mothers can reliably identify individuals by scent alone. Newborn babies quickly learn to recognize the smell of their own mother, and mothers can distinguish the scent of their baby from that of other infants within hours of birth. Your personal scent changes subtly with age, illness, stress, and hormonal shifts, but its underlying signature remains recognizably yours throughout your life. Forensic scientists have explored the possibility of using scent as a biometric identifier, much like a fingerprint or a retinal scan. It is a remarkable thought that your identity is written not only in the lines of your hands, but also in the invisible molecules that drift from your skin. Your sense of smell can distinguish over 1 trillion different odors. For many years, scientists believed the human nose could detect roughly 10,000 different scents. But research published in the 2010s dramatically revised that estimate upward. Using carefully controlled experiments in which volunteers were asked to distinguish between complex mixtures of odor molecules, researchers calculated that the human olfactory system is capable of discriminating more than 1 trillion distinct smells. This number is so large that it dwarfs the number of colors the eye can see or the number of tones the ear can hear. The olfactory system achieves this extraordinary range through a family of roughly 400 different types of olfactory receptor, each tuned to respond to a different set of molecular features. When you inhale a scent, a unique combination of these receptors is activated, creating a pattern that the brain interprets as a specific odor. The number of possible combinations is astronomical, which is why you can tell the difference between the smell of a rose and the smell of jasmine, even though both are complex mixtures of hundreds of volatile compounds. Your nose is one of the most sensitive instruments you possess. It is a quiet chemist, analyzing the air with every breath and reporting its findings to a brain that never stops listening. Holding your nose reduces your ability to taste most flavors. If you have ever eaten while suffering from a heavy cold, you will know that food seems bland and flavorless. This is because much of what we call taste is actually produced by the sense of smell. When you chew food, volatile molecules are released and travel from the back of your mouth up into the nasal cavity through a passage called the nasopharynx. This process, known as retronasal olfaction, allows the olfactory receptors to analyze the food's aroma while it is still in your mouth. The brain then combines this olfactory information with the basic taste signals from the tongue to produce the rich, complex experience of flavor. When you hold your nose or when congestion blocks the nasal passages, the retronasal pathway is shut off, and you are left with only the 5 basic tastes that the tongue can detect on its own: sweet, sour, salty, bitter, and umami. Without smell, chocolate tastes sweet and slightly bitter, but loses all its chocolatey richness. A strawberry becomes merely sweet and acidic. The next time you enjoy a complex meal, remember that your nose is doing at least as much work as your tongue. Flavor is a quiet duet between 2 senses, and neither can perform the full composition alone. Most of what we experience as flavor actually comes from smell. It is one of the most surprising facts about human perception. The tongue can detect only 5 basic taste qualities: sweet, sour, salty, bitter, and umami. Everything else that you perceive as flavor, the difference between a peach and a plum, between basil and mint, between coffee and cocoa, is contributed by the sense of smell. When food is in your mouth, aromatic compounds rise through the back of the throat into the nasal cavity, where hundreds of different olfactory receptors analyze them. The brain seamlessly merges this olfactory information with the basic taste data from the tongue, creating the unified experience of flavor that you associate entirely with eating. Because the merging happens so smoothly, most people are unaware that smell plays such a dominant role. Scientists estimate that somewhere between 70 and 90% of what we perceive as flavor is actually olfactory in origin. This is why professional wine tasters, perfumers, and chefs spend years training their noses. The tongue provides the broad strokes, but the nose paints the fine detail. Flavor, in all its richness, is a quiet masterpiece of sensory integration, a collaboration so seamless that we rarely notice it at all. Some people have thousands more taste buds than average. The number of taste buds on a human tongue varies enormously from person to person. Most adults have somewhere between 2,000 and 10,000 taste buds, but at the extremes, some people have far fewer while others have far more. Those at the high end of the spectrum are sometimes called supertasters. Supertasters tend to experience flavors more intensely than the average person. Bitter foods like Brussels sprouts, dark chocolate, and black coffee can taste almost unbearably strong to them. Sweet and salty flavors may also be amplified. The difference is largely genetic. A specific gene influences the density of taste buds on the tongue, and those who inherit certain variants end up with a significantly higher concentration of the tiny sensory structures called fungiform papillae. Women are more likely to be supertasters than men, and the trait appears to be more common in certain populations. Being a supertaster is not necessarily an advantage. Some supertasters avoid healthy but bitter vegetables, while others find strongly flavored foods overwhelming. It is simply a different way of experiencing the world, a reminder that the same plate of food does not taste the same to everyone. Perception is personal, shaped by the quiet biology of each individual tongue. Newborn babies instinctively prefer sweet tastes from birth. Within hours of being born, a baby will smile and relax when given a drop of sugar water and grimace or turn away from something bitter. This preference for sweetness is not learned. It is hardwired into the human nervous system, a biological inheritance shaped by millions of years of evolution. In the natural world, sweet tastes are generally associated with energy-rich foods like ripe fruit, while bitter tastes are often associated with toxins and poisons. By building a preference for sweetness into the newborn brain, evolution has equipped babies with an immediate guide to safe nutrition, long before they have any understanding of the world around them. The aversion to bitterness is equally innate and equally protective. These preferences soften and change as a child grows. Exposure to a wide variety of flavors during early life can expand the range of foods a person enjoys as an adult. But the underlying bias towards sweetness and away from bitterness never disappears entirely. Even as adults, most humans retain a fondness for sweet foods that can be traced back to this ancient survival mechanism. It is a quiet instinct written into the very first moments of life, a taste preference older than language itself. The burning sensation from spicy food is actually a pain response. When you eat a hot chili pepper, the burning feeling in your mouth is not a taste at all. It is a pain signal. The active compound in chili peppers, capsaicin, binds to a specific receptor on nerve cells called TRPV1. This receptor is the same one that detects actual physical heat. When capsaicin activates it, the receptor sends a signal to the brain that is interpreted as a burning sensation, even though no real damage is occurring. Your mouth is not actually on fire. Your nervous system simply cannot tell the difference between capsaicin and genuine heat. This is why drinking cold water provides temporary relief, and why dairy products, which contain a protein called casein that binds to capsaicin and washes it away from the receptors, are more effective at easing the burn. Interestingly, birds lack the TRPV1 receptor that responds to capsaicin, so they can eat chili peppers without any discomfort. This is thought to be an evolutionary advantage for the plant, since birds disperse seeds more effectively than mammals. The love of spicy food is a learned behavior, a quiet thrill-seeking of the nervous system. Each bite is a gentle trick played on the brain, a harmless pain that many people learn to enjoy. Your tongue detects 5 basic tastes, but your brain creates thousands. The human tongue is equipped with receptors for 5 basic taste qualities: sweet, sour, salty, bitter, and umami. These 5 categories represent the fundamental building blocks of taste, each one providing specific information about the nutritional content or potential danger of what you are eating. Sweet signals energy. Salty indicates minerals. Sour warns of acidity. Bitter flags possible toxins. Umami detects protein-rich foods. But the experience of eating a meal involves far more than these 5 channels. The brain combines basic taste information with input from the sense of smell, the texture detected by touch receptors in the mouth, the temperature of the food, and even its visual appearance and the sounds it makes when chewed. From these raw inputs, the brain constructs the thousands of distinct flavor experiences that make up your culinary world. The difference between a ripe mango and a green apple, between dark chocolate and milk chocolate, between fresh bread and stale bread, all emerge from the brain's ability to integrate and interpret multiple sensory streams simultaneously. The tongue provides the alphabet, but the brain writes the poetry. Flavor is not found on the tongue. It is composed in the mind, a quiet act of sensory creativity repeated with every bite. Scientists believe humans may have a sixth taste for detecting fats. For decades, the textbook answer was that humans possess 5 basic tastes. But in recent years, a growing body of research has suggested that there may be a sixth. Scientists have identified receptors on the tongue that appear to respond specifically to fatty acids, the building blocks of dietary fats. This proposed sixth taste has been tentatively named oligustus. In controlled experiments, subjects have been able to detect the presence of fatty acids in solutions that have been stripped of all texture, aroma, and visual cues, suggesting that the response is a genuine taste rather than a tactile or olfactory perception. The sensation of oligustus is not the creamy richness that most people associate with fatty foods. Isolated fatty acids actually taste rather unpleasant, which may serve as a warning against consuming rancid or spoiled fats. The pleasant experience of fatty food likely comes from the combination of oligustus with other tastes, textures, and aromas. If confirmed, the existence of a fat taste would make evolutionary sense. Fats are the most energy-dense nutrient available, and having a dedicated receptor to detect them would help guide dietary choices. The science is still developing, but the possibility of a sixth taste is a quiet expansion of our understanding of human perception. Cats cannot taste sweetness at all. Domestic cats, along with all other members of the cat family, lack a functional gene for one of the 2 proteins that form the sweet taste receptor. This means that they are unable to detect sweetness in any form. While a human might find a ripe peach or a spoonful of honey irresistible, a cat would perceive no sweetness whatsoever. This genetic mutation is thought to have occurred early in the evolution of the cat lineage, long before the divergence of domestic cats from their wild ancestors. It makes evolutionary sense. Cats are obligate carnivores, meaning they are biologically adapted to a diet consisting almost entirely of meat. They have no nutritional need for sugars or carbohydrates, so the ability to detect sweetness would offer them no survival advantage. Over time, the gene degraded without consequence. The loss of the sweet receptor is just one of many ways in which the cat's sensory world differs from our own. They possess extraordinary night vision, a keen sense of hearing, and a sense of smell far more sensitive than ours. But the world of sweetness is entirely closed to them. It is a gentle reminder that every species experiences the world through its own unique sensory lens, shaped by the demands of its particular way of life. Many fish can taste with their entire bodies. While humans confine their sense of taste to the tongue and the back of the throat, many species of fish have taste receptors distributed across their entire body surface. Catfish are perhaps the most extreme example. They are covered in hundreds of thousands of taste buds, concentrated especially on their barbels, the whisker-like structures near their mouths, but also scattered across their fins, sides, and even their tails. This gives the catfish the ability to taste the water around it in every direction simultaneously, an invaluable adaptation for a bottom-dwelling fish that often hunts in murky, low-visibility environments. By tasting the chemicals dissolved in the water, a catfish can detect the presence of food, identify potential predators, and navigate its surroundings without relying on sight. Other fish, including carp and some species of goby, also possess external taste receptors, though generally in smaller numbers. This whole-body tasting ability is a vivid demonstration of how the senses can be distributed across the body in ways that are radically different from the human norm. For these fish, the boundary between tasting and touching is blurred. Their entire skin is a sensory organ, quietly sampling the chemical composition of every drop of water that passes over them. Octopuses taste and feel simultaneously through their arms. Each of an octopus's 8 arms is lined with hundreds of suckers, and each sucker is packed with both tactile and chemical receptors. This means that when an octopus touches something, it is simultaneously feeling its texture and tasting its chemical composition. The octopus does not need to bring food to its mouth to evaluate it. It can taste an object the moment it makes contact. This dual sensory capability is controlled in part by the arms themselves. Each arm contains a dense cluster of neurons that can process sensory information and initiate simple responses independently of the central brain. An octopus arm that has been severed will still respond to touch and withdraw from harmful stimuli for a period of time. This extraordinary distribution of sensory processing across the body gives the octopus an almost alien form of awareness. It can explore a coral reef, a rocky crevice, or the surface of a shell with a richness of information that combines what humans would experience as 2 entirely separate senses. For the octopus, touch and taste are not different experiences. They are a single, unified way of knowing the world, a quiet intimacy with its surroundings that we can only begin to imagine. Butterflies taste the world through receptors on their feet. When a butterfly lands on a leaf or a flower, it is doing far more than resting. Its feet contain chemoreceptors, specialized sensory cells that detect the chemical composition of the surface it has touched. This allows the butterfly to taste its environment the instant it makes contact. For a female butterfly searching for a place to lay her eggs, this ability is essential. She must identify the specific host plant that her caterpillars will need to eat after hatching. By tasting the leaf with her feet, she can determine in a fraction of a second whether the plant is the right species and whether it is healthy enough to support her offspring. If it is not, she moves on. If it is, she deposits her eggs. This foot-tasting behavior is remarkably efficient. It eliminates the need for the butterfly to use its proboscis to sample every surface, saving energy and time. The receptors on the feet can detect sugars, salts, and other compounds with impressive sensitivity. It is a beautiful example of how natural selection tailors the placement of sensory organs to the specific needs of each species. For the butterfly, the world is tasted 1 footstep at a time, a quiet survey carried out with every gentle landing. The sense of touch begins developing before birth. Touch is the first of the senses to develop in a human embryo. By roughly 8 weeks of gestation, the developing fetus begins to respond to tactile stimulation around the mouth and cheeks. Over the following weeks, sensitivity spreads progressively across the body, reaching the palms of the hands, the soles of the feet, and eventually the entire skin surface. Long before a baby can see, hear, or smell the outside world, it is already experiencing touch within the womb. The gentle pressure of the amniotic fluid, the movements of its own limbs, and the rhythmic pulsation of the mother's heartbeat all provide a rich landscape of tactile stimulation. This early sensory input is thought to play an important role in the development of the nervous system, helping to refine the neural pathways that will later process more complex tactile information. By the time of birth, the newborn's sense of touch is already highly developed. Babies respond immediately to the warmth and pressure of being held, and skin-to-skin contact with the mother has been shown to regulate heart rate, body temperature, and stress hormones. Touch is the first sense to arrive and the last to fade. It is the quiet foundation upon which all other sensory experience is built. Your skin contains more than 20 types of nerve endings. The human skin is not a single uniform sensory surface. It is a mosaic of specialized receptors, each tuned to detect a different aspect of the physical world. Meissner's corpuscles respond to light touch and are concentrated in the fingertips and lips. Pacinian corpuscles detect deep pressure and vibration and are found in the palms and the soles of the feet. Merkel cells sense sustained pressure and fine texture. Ruffini endings respond to stretching of the skin. Free nerve endings detect pain and temperature. Together, these and other receptor types give the skin the ability to detect an extraordinary range of stimuli, from the lightest brush of a feather to the crushing force of a heavy object, from the gentle warmth of sunlight to the sharp sting of a needle. The density and distribution of these receptors vary across the body. The fingertips, lips, and tongue have the highest concentration, which is why these areas are the most sensitive. The back and the legs have fewer receptors and are correspondingly less discriminating. This uneven distribution reflects the evolutionary importance of sensitive hands and mouths for manipulating objects, finding food, and communicating socially. Your skin is a vast, quiet sensory organ, mapping the physical world with extraordinary precision across every centimeter of your body. Some individuals can feel textures others cannot even detect. Human tactile sensitivity varies significantly from person to person. At one end of the spectrum, some individuals can detect the difference between surfaces that differ by only a few microns in texture, a level of precision that rivals or exceeds the resolution of a fingertip-mounted scientific instrument. At the other end, some people require much larger differences before they can tell two surfaces apart. This variation is influenced by several factors. The density of Meissner's corpuscles and Merkel cells in the fingertips plays a major role. People with smaller fingers tend to have receptors packed more closely together, which is one reason why women, on average, have slightly greater tactile acuity than men. Age also matters. The number of touch receptors in the skin declines with age, which is why older adults sometimes struggle with tasks that require fine tactile discrimination. Practice can sharpen the sense of touch as well. Braille readers, musicians, and craftspeople who work with their hands develop heightened tactile sensitivity over time as the brain devotes more processing power to the fingertips. The world of texture is richer for some than for others, a quiet inequality in perception that most people never notice. A rare condition links touch and emotion so strongly it becomes overwhelming. Mirror touch synesthesia is a condition in which a person physically feels the sensations that they observe in another person. If someone with this condition sees another person being touched on the cheek, they feel a corresponding sensation on their own cheek. If they watch someone stub a toe, they may experience a flash of pain in their own foot. This is not ordinary empathy. It is a genuine sensory experience, confirmed by brain imaging studies that show activation of the somatosensory cortex, the same region that processes direct physical touch. The condition is thought to arise from an overactive mirror neuron system, the network of brain cells that fires both when you perform an action and when you observe someone else performing the same action. In most people, this system generates only a subtle empathic response. In those with mirror touch synesthesia, it produces a full sensory experience. Living with this condition can be deeply challenging. Crowded places become overwhelming because every observed touch is felt. Watching someone in pain can be physically distressing, yet many who have it report that it also deepens their connection to others. It is a quiet intensification of the bond between human beings, a sensory bridge that makes the boundary between self and other remarkably thin. Your body senses heat and cold through entirely separate receptors. The ability to detect temperature is not a single sense, but two distinct systems operating in parallel. Warmth is detected by one set of nerve endings, and cold is detected by an entirely different set. The warm receptors respond to temperatures above the neutral range of the skin, while the cold receptors respond to temperatures below it. Each type of receptor contains specialized ion channels that open in response to specific temperature ranges, generating electrical signals that travel to the brain. The cold-sensitive channels are activated at lower temperatures and are closely related to the receptor that responds to menthol, which is why peppermint feels cool on the skin even at room temperature. The warm-sensitive channels are activated at higher temperatures and are related to the capsaicin receptor that produces the burning sensation from chili peppers. If both types of receptor are stimulated simultaneously, as happens when you touch an object that is warm in some areas and cold in others, the brain can be tricked into perceiving a burning sensation even when no damaging heat is present. This phenomenon, known as the thermal grill illusion, reveals just how actively the brain constructs the experience of temperature from the raw data of its receptors. It is a quiet reminder that sensation is always an interpretation. The lips are among the most sensitive parts of the human body. The skin of the lips contains an exceptionally high density of touch receptors, making them one of the most tactily sensitive areas of the entire body. This sensitivity is reflected in the brain's somatosensory cortex, the region that processes touch. In the cortical map of the body, the lips are represented by a disproportionately large area relative to their physical size, a feature illustrated by the famous sensory homunculus. The reason for this heightened sensitivity is rooted in evolution. The lips play a critical role in feeding, especially in infancy when a baby must locate and latch onto the breast. They also play a central role in speech, helping to shape the sounds that form language. Beyond these practical functions, the sensitivity of the lips makes them important in social bonding and emotional communication. A kiss, a gentle touch against the lips, or even the feel of a warm drink can trigger a cascade of sensory and emotional responses. The concentration of nerve endings in the lips also makes them effective at evaluating the texture and temperature of food before it enters the mouth. Every function of the lips, from nursing to speaking to expressing affection, depends on this extraordinary sensitivity. It is a quiet gift of biology, a concentration of awareness in one of the most expressive parts of the human face. Blind individuals often develop extraordinarily precise texture perception. When the visual system is absent from birth or lost early in life, the brain does not leave the visual cortex idle. Instead, through a process known as cross-modal plasticity, the areas of the brain that would normally process visual information are recruited to process input from other senses, particularly touch and hearing. This neural reorganization allows many blind individuals to develop tactile abilities that significantly exceed those of sighted people. Experienced Braille readers, for example, can distinguish raised dots separated by fractions of a millimeter, reading at speeds that rival sighted reading. Studies have shown that the visual cortex of proficient Braille readers becomes active during tactile tasks, confirming that these brain areas have been repurposed. The enhanced tactile perception is not limited to Braille. Blind individuals often excel at recognizing objects by touch, detecting subtle differences in texture, and navigating complex environments using tactile cues. This remarkable adaptability of the brain demonstrates that the senses are not rigidly compartmentalized. The brain is a flexible organ that allocates its resources based on the demands placed upon it. When one sensory channel is closed, the others can expand to fill the space. It is a quiet testament to the resilience and ingenuity of the human nervous system. The sensation of being ticklish evolved for bonding and self-defense. Ticklishness is a curious sensation that most people take for granted, but it has deeper evolutionary roots than you might expect. Scientists distinguish between two types. Nismesis is the light, irritating sensation caused by something crawling on the skin, such as an insect. It triggers an automatic brushing or scratching response designed to remove potential threats. Gargulses is the heavier tickling sensation that occurs in specific body areas like the armpits, sides, and feet, and it typically provokes laughter and squirming. Interestingly, you cannot tickle yourself. The brain's cerebellum predicts the sensory consequences of your own actions and cancels out the tickle response. This suggests that ticklishness evolved specifically as a response to unexpected external contact. The laughter triggered by gargulses is thought to serve a social bonding function, particularly between parents and children. Tickle play encourages physical closeness and teaches young children about trust, boundaries, and social interaction. The vulnerability of the most ticklish areas, the neck, the ribs, the belly, may also reflect an ancient defensive response, training the body to protect its most exposed regions. Ticklishness is a quiet intersection of defense and delight, a quirk of the nervous system that connects safety and affection. Phantom limb sensations reveal how touch is mapped inside the brain. After an arm or a leg is amputated, many people continue to feel vivid sensations in the missing limb. They may feel itching, warmth, pressure, or even pain in a hand or foot that no longer exists. These phantom limb sensations demonstrate that the experience of touch is not generated in the limbs themselves, but in the brain. The somatosensory cortex contains a detailed map of the entire body, with each region of the cortex corresponding to a specific part of the body's surface. When a limb is lost, the corresponding area of the cortex does not simply go silent. Instead, it can be invaded by signals from neighboring regions of the body map. This is why touching the face or the remaining stump can sometimes trigger sensations that seem to come from the missing limb. The brain's body map is remarkably persistent. It was built during development and reinforced over years of sensory experience, and it does not simply erase itself when the limb is removed. Phantom limb pain, which can be severe and debilitating, is one of the most challenging consequences of this neural persistence. Treatments that use mirrors or virtual reality to provide visual feedback to the brain have shown promise in relieving this pain. Phantom limbs are a quiet and poignant reminder that sensation lives not in the body, but in the mind. Your brain constantly predicts what you are about to feel. The brain does not passively wait for sensory information to arrive. It actively generates predictions about what it expects to feel, see, and hear at every moment. When you reach out to pick up a cup of coffee, your brain has already predicted the weight, the warmth, and the smooth texture of the ceramic before your fingers make contact. If the cup turns out to be heavier than expected, or hotter or rougher, the mismatch between prediction and reality generates a surprise signal that captures your attention. This predictive system is remarkably efficient. By precomputing what is likely to happen, the brain reduces the amount of processing needed for each new sensory event. Only the differences between prediction and reality, the so-called prediction errors, require full attention. Everything that matches the prediction is processed automatically and quietly. This is why you can type on a keyboard or button a shirt without thinking about each individual sensation. Your brain has learned the expected patterns and handles them in the background. Predictive processing is thought to underlie not only touch, but all sensory experience. The world you perceive at any given moment is not a raw recording. It is a carefully constructed model, constantly updated by the quiet interplay between expectation and incoming data. Sound vibrations can be felt directly through the skin. Sound is a mechanical wave, a series of pressure changes that travel through a medium such as air, water, or solid material. While we normally perceive these vibrations through the specialized structures of the ear, the skin is also capable of detecting them directly. Low-frequency sounds, in particular, can be felt as vibrations in the chest, the abdomen, and the palms of the hands. This is why standing near a loudspeaker during a concert produces a physical sensation throughout the body, not just a sound in the ears. The skin's ability to detect vibration is mediated primarily by Pacinian corpuscles, the deep pressure receptors that are also responsible for detecting texture and fine vibration through touch. Research has shown that tactile perception of sound can enhance and modify auditory perception. When sound and tactile vibration arrive together, the brain integrates them into a single, richer experience. This multisensory integration is the basis for technologies that translate sound into vibration for people with hearing loss, allowing them to perceive rhythm, pitch, and even speech through their skin. The body is not just a listener. It is a resonator, a quiet instrument that vibrates in sympathy with the sounds around it, connecting you to the physical reality of every wave that passes through the air. Deaf individuals often experience music through vibration. For many people who are deaf or severely hard of hearing, music is not a silent experience. It is a tactile one. The low-frequency vibrations produced by drums, bass instruments, and amplified speakers can be felt through the floor, the furniture, and the body itself. Many deaf musicians and music lovers describe a rich and meaningful experience of rhythm, tempo, and even emotional content through these physical vibrations alone. The famous percussionist Evelyn Glennie, who has been profoundly deaf since childhood, performs barefoot so that she can feel the vibrations of the instruments through the stage floor. Her career demonstrates that musical perception is not confined to the ears. The brain regions that process rhythm and temporal patterns can be activated by vibration as much as by sound. Technological advances have expanded access further. Vibrotactile devices that translate audio frequencies into patterns of vibration worn on the skin allow deaf individuals to experience music with increasing nuance. Research into these devices has shown that with practice, users can learn to distinguish pitch, rhythm, and even timbre through touch alone. Music, it turns out, is not exclusively an auditory phenomenon. It is a physical one, a quiet resonance that the body can feel even when the ears cannot hear. Your ears never truly stop hearing, even while you sleep. When you fall asleep, your eyes close, your muscles relax, and your conscious awareness fades, but your ears remain open. Unlike the eyes, which have lids that can block incoming light, the ears have no physical mechanism for shutting out sound. The auditory system continues to receive and process sounds throughout the night, even during the deepest stages of sleep. What changes is how the brain responds to that information. During sleep, the thalamus, a relay station deep within the brain, acts as a gatekeeper, filtering out most routine sounds and allowing only significant or unexpected stimuli to reach conscious awareness. This is why you can sleep through the steady hum of traffic or the gentle whir of a fan, but be jolted awake by the sound of your name or a sudden crash. The brain is continuously evaluating the auditory environment, deciding moment by moment what is safe to ignore and what demands a response. This selective filtering is an ancient survival mechanism. In the wild, the ability to detect threatening sounds during sleep could mean the difference between life and death. Even now, in the safety of your bedroom, your ears are standing watch. They are patient sentinels, quietly monitoring the world around you while the rest of your body rests. The quietest sound you can hear moves your eardrum less than an atom. The human ear is an instrument of astonishing sensitivity. At the threshold of hearing, the faintest sound that a healthy young person can detect causes the eardrum to vibrate by a distance smaller than the diameter of a single hydrogen atom. This is a movement so tiny that it approaches the scale of thermal molecular motion, the random jiggling of molecules caused by heat. If the ear were any more sensitive, it would begin to pick up the noise of its own molecules vibrating, rendering hearing useless. The fact that evolution has pushed the sensitivity of the ear so close to this physical limit is remarkable. The ear achieves this through a chain of mechanical amplification. Sound waves are funneled by the outer ear, concentrated by the eardrum, and amplified by the three tiny bones of the middle ear, the malleus, incus, and stapes. The stapes, the smallest bone in the human body, transmits the vibrations into the fluid-filled cochlea, where they are converted into electrical signals by the hair cells. This entire chain of amplification is exquisitely tuned. The ear can detect sounds ranging from the faintest whisper to the roar of a jet engine, a dynamic range that spans more than a trillion-fold difference in sound pressure. It is a quiet miracle of engineering, refined by hundreds of millions of years of evolution. Your brain locates sounds using differences smaller than a microsecond. The ability to determine where a sound is coming from depends on the brain's capacity to detect incredibly small differences in the time it takes for sound to reach each ear. A sound coming from the left arrives at the left ear a fraction of a second before it reaches the right ear. The brain measures this interaural time difference and uses it to calculate the direction of the source. For sounds coming from directly to one side, the time difference is roughly 600 to 700 microseconds. But the brain can detect differences as small as 10 to 20 microseconds, allowing it to localize sounds with remarkable precision. This is not a simple calculation. The brain also uses differences in loudness between the two ears and the way the shape of the outer ear modifies the sound to determine direction and distance. All of these cues are processed simultaneously and integrated into a spatial map of the auditory environment. This ability was crucial for survival in the ancestral environment, where detecting the direction of a predator or a calling companion could mean the difference between safety and danger. Even now, in the quiet of your room, your brain is effortlessly tracking the direction of every sound that reaches your ears. It is a feat of neural computation so precise that no human-made system has yet matched it. Earworms are songs that loop in the brain like auditory hallucinations. Almost everyone has experienced the sensation of a song playing on repeat inside their head, long after the actual music has stopped. These involuntary musical images, commonly known as earworms, are among the most widespread quirks of human auditory perception. Research suggests that earworms are triggered most often by songs that are simple, repetitive, and recently heard. They tend to involve fragments rather than complete songs, typically a chorus or a catchy melodic hook that loops endlessly. The phenomenon is related to the way the brain processes and stores auditory information. When you hear a familiar song, your auditory cortex activates to process it. Sometimes, the neural circuits that encode that fragment become spontaneously reactivated, producing the sensation of hearing the music even in silence. Earworms are more common during periods of low cognitive load, such as when you are walking, showering, or trying to fall asleep. Some people find them pleasant, while others find them maddening. Interestingly, actively listening to the complete version of the song or engaging in a mentally demanding task can sometimes break the loop. Earworms are not a sign of any disorder. They are simply a byproduct of a brain designed to remember and predict patterns in sound. They are a quiet echo of music that refuses to fade. Bats process echoes hundreds of times faster than humans can. Echolocation is the remarkable ability to navigate and hunt by emitting high-frequency sound pulses and interpreting the echoes that bounce back from surrounding objects. Many species of bat rely on this system as their primary means of sensing the environment, particularly in the darkness of night when vision is of limited use. A bat emits ultrasonic calls, often at frequencies far above the range of human hearing, and then listens for the returning echoes with extraordinary precision. By analyzing the time delay, the frequency shift, and the intensity of each echo, the bat can determine the size, shape, distance, direction, and even the texture of an object in its path. The speed of this processing is staggering. Some bats can emit and process hundreds of calls per second, adjusting their call rate and frequency in real time as they close in on a flying insect. The entire sequence, from call to echo to neural interpretation to motor correction, takes only a few milliseconds. For comparison, the human brain requires tens of milliseconds simply to register a basic auditory stimulus. Bat echolocation is one of the most sophisticated sensory systems in the animal kingdom. It is a quiet concert of sound and silence, played out in frequencies too high for human ears to witness. Elephants can hear sounds below the range of human hearing. Elephants communicate using infrasound sound waves with frequencies below 20 Hz, which is the lower limit of the typical human hearing range. These low-frequency calls can travel vast distances across the African savanna, carried further by the ground than by the air alone. Research has shown that elephants can detect these rumbles from distances of several kilometers, allowing herds separated by great distances to coordinate their movements, warn one another of danger, and locate water sources. The elephant's ability to detect infrasound is aided not only by its large ears, but also by specialized cells in its feet that are sensitive to ground vibrations. When another elephant vocalizes at very low frequencies, the sound waves travel through the earth and are picked up by the listening elephant's feet and lower legs, where they are transmitted through bone to the inner ear. This dual reception system, combining airborne and ground-borne sound, gives the elephant an acoustic awareness that extends far beyond what any human could perceive. Elephants essentially hear with their whole bodies. It is a quiet, deep conversation carried on frequencies too low for us to notice, connecting individuals across the vast and open landscape. Whale songs can travel thousands of miles through the ocean. The ocean is a remarkably efficient medium for the transmission of sound. Water is denser than air, and sound waves travel through it roughly 4 times faster. At certain depths, a layer of water known as the SOFAR channel acts as a natural waveguide, trapping sound waves and allowing them to propagate across enormous distances with very little loss of energy. Humpback whales, fin whales, and blue whales exploit this channel to communicate across entire ocean basins. The low-frequency calls of a blue whale, the largest animal ever to have lived, can travel thousands of kilometers through the deep ocean. These calls serve a variety of purposes, from attracting mates to maintaining contact between individuals separated by hundreds of miles of open water. The songs of humpback whales are among the most complex vocalizations in the animal kingdom, consisting of long, structured sequences of sounds that evolve over time. All males in a given population sing roughly the same song, and the song changes gradually from year to year. The deep ocean is not the silent world it was once thought to be. It is filled with the quiet music of whales, a vast, slow conversation carried on sound waves that cross continents beneath the surface of the sea. Owls can locate prey in complete darkness, using hearing alone. The barn owl is one of nature's most precise auditory hunters. It can catch a mouse in complete darkness, relying entirely on the faint sounds the prey makes as it moves through leaf litter or grass. This extraordinary ability is the result of several remarkable adaptations. The owl's facial disc, the flat, heart-shaped arrangement of feathers around its face, acts as a parabolic reflector, funneling sound toward the ear openings. In many owl species, the ears are positioned asymmetrically on the skull, with one slightly higher than the other. This asymmetry allows the owl to determine the vertical position of a sound source, as well as its horizontal direction. By combining the information from both ears, the owl can pinpoint the exact three-dimensional location of its prey with remarkable accuracy. Laboratory experiments have shown that barn owls can strike a target in total darkness with an error of less than a single degree. The neural circuitry that supports this ability is concentrated in a specialized region of the midbrain that creates a detailed spatial map of the auditory environment. The owl's hearing is so finely tuned that it can detect and locate sounds that a human ear would never notice. It is the quiet precision of a hunter whose world is built entirely from sound. Your sense of balance lives deep within your inner ear. The vestibular system, which governs your sense of balance and spatial orientation, is located in the inner ear, tucked away inside the temporal bone of the skull. It consists of 3 semicircular canals and 2 otolith organs, the utricle and the saccule. The semicircular canals are arranged at right angles to one another, each one filled with a fluid called endolymph. When you turn your head, the fluid in the corresponding canal shifts, bending tiny hair cells that line the inside of the canal. These hair cells convert the mechanical movement into electrical signals that are sent to the brain, informing it of the direction and speed of the rotation. The otolith organs, by contrast, detect linear acceleration and the pull of gravity. They contain tiny crystals of calcium carbonate embedded in a gel-like membrane. When you tilt your head or move forward, the crystals shift under the influence of gravity or inertia, stimulating the hair cells beneath them. Together, these structures provide a continuous stream of information about your position and movement in space. The brain integrates this vestibular data with visual input and proprioceptive signals from the muscles and joints to maintain your balance and coordination. It is all happening quietly, deep inside your skull, without any conscious effort on your part. Spinning makes you dizzy because fluid in your ears keeps moving. When you spin around rapidly and then suddenly stop, the world appears to continue spinning for several seconds. This sensation of dizziness, known as vertigo, is caused by the fluid dynamics of the semicircular canals in your inner ear. While you are spinning, the endolymph fluid inside the canals eventually catches up with the rotation and begins moving at the same speed as your head. But when you stop abruptly, your head is stationary while the fluid continues to move for a few moments due to its inertia. The hair cells lining the canals detect this ongoing fluid movement and send signals to the brain indicating that you are still rotating, even though you are not. The brain receives conflicting information. The vestibular system says you are moving, but the visual system says you are still, and the resulting mismatch produces the disorienting sensation of dizziness. Nausea can follow if the conflict is severe, because the brain interprets sensory mismatch as a possible sign of poisoning, triggering a protective nausea response. This is also the mechanism behind motion sickness. The vertigo gradually fades as the fluid in the canals comes to rest and the sensory signals align once again. It is a quiet demonstration of the brain's dependence on consistent sensory information to construct a stable perception of the world. Astronauts experience confusion when gravity no longer guides balance. On Earth, the vestibular system relies heavily on the constant downward pull of gravity to determine which way is up. The otolith organs in the inner ear use the weight of tiny calcium carbonate crystals to sense the direction of gravitational pull, providing the brain with a reliable reference frame for orientation. In the microgravity environment of space, this reference frame disappears. The crystals no longer settle downward, and the otolith organs can no longer distinguish between up and down. Astronauts arriving in orbit for the first time often experience a condition known as space adaptation syndrome, which includes nausea disorientation, and difficulty coordinating movements. The brain must learn to ignore the now unreliable vestibular signals and rely more heavily on visual cues and proprioception. This adaptation typically takes several days to a week. Some astronauts report that simply closing their eyes in microgravity produces a profound sense of disconnection, as though they have lost all awareness of their body's position in space. Upon returning to Earth, the readaptation to gravity can be equally challenging. The vestibular system must recalibrate itself to the pull of gravity after weeks or months of functioning without it. It is a quiet reminder of how deeply our sense of self depends on the invisible partnership between our inner ears and the force that holds us to the planet. Some fish sense movement through pressure-detecting lateral lines. Many fish and some aquatic amphibians possess a sensory system that has no counterpart in humans. The lateral line system consists of a series of specialized sensory organs called neuromasts, arranged in a line along each side of the body and over the head. Each neuromast contains hair cells similar to those found in the human inner ear, and they respond to changes in water pressure and current flow. When another animal moves nearby, it creates tiny pressure waves in the water. The lateral line detects these waves and transmits the information to the brain, allowing the fish to build a detailed picture of its immediate surroundings without relying on sight. This is invaluable in murky water or at night, when visibility is poor. The lateral line also helps fish detect obstacles, maintain position in a current, and coordinate schooling behavior. In a school of hundreds of fish, each individual uses its lateral line to monitor the movements of its neighbors, allowing the entire group to turn, accelerate, and decelerate in near-perfect synchrony. Predatory fish use the same system to detect the faint pressure waves generated by the movements of their prey. The lateral line is a quiet sense, invisible from the outside, but it gives its possessors an awareness of the water world that is extraordinarily detailed and immediate. Human eyes detect only a tiny fraction of the electromagnetic spectrum. The electromagnetic spectrum stretches from radio waves with wavelengths of kilometers to gamma rays with wavelengths smaller than an atom. It encompasses an enormous range of energy and frequency. Yet the human eye is sensitive to only a vanishingly small sliver of this spectrum, a band of wavelengths from roughly 380 nanometers to 700 nanometers. This is what we call visible light. Everything outside this narrow window, including radio waves, microwaves, infrared radiation, ultraviolet light, X-rays, and gamma rays, is completely invisible to us. The reason for this limitation is rooted in biology. The photoreceptor cells in the retina contain light-sensitive pigments that respond only to photons within the visible range. Evolution shaped these pigments to match the wavelengths of light most abundantly emitted by the sun and most readily transmitted through water and the atmosphere. For the purposes of survival, this narrow band provides all the information our ancestors needed. But it is humbling to consider how much of the electromagnetic universe lies entirely beyond our perception. The warm glow of infrared, the ultraviolet patterns on flowers, the radio hum of distant galaxies, all of it exists around you at every moment, silent and unseen. Your eyes reveal a beautiful world, but it is only a quiet fraction of the reality that surrounds you. Bees can see ultraviolet patterns invisible to human vision. The visual world of a honeybee is startlingly different from our own. While human eyes contain 3 types of color receptor, tuned to red, green, and blue light, bee eyes contain receptors tuned to green, blue, and ultraviolet. This means that bees can see ultraviolet light, which is completely invisible to the human eye, but they cannot see red. Many flowers that appear plain white or yellow to us display striking patterns when viewed in ultraviolet, including bold stripes, concentric rings, and bullseye targets that point directly toward the nectar. These patterns, known as nectar guides, are invisible to humans but perfectly visible to the bees for which they evolved. The coevolution of flowers and their pollinators has produced a visual language written in wavelengths that we can only detect with specialized cameras. For a bee, a meadow is not simply a field of color. It is an intricate map of navigational cues, each flower broadcasting its location and its reward in a code that only its intended audience can read. This difference in visual perception is a vivid reminder that the world does not have one appearance. Every species sees a different version of reality, shaped by the particular wavelengths its eyes are built to detect. The bee's world is a quiet, ultraviolet masterpiece. Certain snakes perceive heat using infrared-sensitive pits. Pit vipers, pythons, and boas possess a remarkable sensory adaptation that allows them to detect infrared radiation emitted by warm-blooded animals. In pit vipers, this ability is mediated by a pair of small openings, called pit organs, located between the eye and the nostril on each side of the head. Each pit organ contains a thin membrane densely packed with heat-sensitive nerve endings. When infrared radiation from a nearby warm body strikes the membrane, it causes a slight rise in temperature that is detected by the nerve cells and transmitted to the brain. The brain processes this information alongside visual data, creating a composite image that combines sight and thermal information. This allows the snake to detect and strike at warm prey with remarkable accuracy, even in complete darkness. The sensitivity of the pit organs is extraordinary. They can detect temperature differences of as little as a fraction of a degree Celsius. For a nocturnal predator hunting small rodents that move through dense vegetation at night, this infrared sense provides a decisive advantage. The snake perceives the world in a way that includes an entire dimension of experience invisible to humans. It sees the warmth radiating from living bodies, a quiet glow that reveals the presence of life even in the deepest darkness. Mantis shrimp possess up to 16 types of color receptors. The mantis shrimp is one of the most visually extraordinary creatures on the planet. While humans have 3 types of color receptor, or cone, in their retinas, the mantis shrimp has up to 16. These receptors are sensitive to a vast range of wavelengths, from deep ultraviolet to far red, and they include dedicated receptors for polarized light. At first glance, this might suggest that the mantis shrimp sees a richer world of color than any other animal. However, research has shown that the mantis shrimp's color vision works differently from ours. Rather than blending signals from different receptor types to create subtle color distinctions, the mantis shrimp appears to use each receptor type as an independent channel, creating a rapid scanning system that recognizes color categories rather than mixing them. This approach sacrifices fine color discrimination in favor of speed, allowing the mantis shrimp to identify objects very quickly in the dynamic and visually complex environment of a coral reef. The mantis shrimp's eyes are mounted on mobile stalks that can move independently, giving each eye a full field of trinocular vision. The result is a visual system unlike any other in the animal kingdom. It is a quiet marvel of evolutionary innovation, a reminder that there are many possible solutions to the challenge of seeing the world. Birds perceive colors that do not exist in human experience. Most birds have 4 types of color receptor in their retinas, compared to the 3 that humans possess. The 4th cone type is sensitive to ultraviolet light, giving birds access to a dimension of color that lies entirely outside human perception. This tetrachromatic vision means that birds can see combinations of wavelengths that the human brain cannot represent. What we perceive as a single shade of yellow, a bird might see as 2 distinct colors, one reflecting ultraviolet and one not. The plumage of many bird species, which appears plain or subtly patterned to human eyes, is revealed under ultraviolet light to contain bold markings, iridescent patches, and dramatic patterns used in mate selection and species recognition. Male blue tits, for example, have ultraviolet reflective crown feathers that are invisible to humans but highly conspicuous to other blue tits. Fruit and foliage also look different to tetrachromatic eyes. Ripe berries that blend into the background for a human may stand out vividly to a bird. The avian visual world is richer, more colorful, and more detailed than anything we can imagine. It is a quiet privilege that birds carry with them, a view of the world painted in colors that have no names in any human language. Some deep-sea creatures generate their own light to see in darkness. In the deep ocean, below the reach of sunlight, many organisms produce their own light through a process called bioluminescence. This living light is generated by chemical reactions, typically involving a molecule called luciferin and an enzyme called luciferase. The organisms that produce bioluminescence use it for a variety of purposes. Some deep-sea fish use light organs located near their eyes to illuminate the dark water in front of them, functioning as biological headlamps. Others use bioluminescence to attract prey. The anglerfish, for example, dangles a luminescent lure from a modified fin spine, drawing smaller fish toward its waiting jaws. Some species use flashes of light to startle or confuse predators, while others produce bioluminescent clouds that act as a smokescreen during escape. Communication is another important function. Many deep-sea organisms use species-specific patterns of light to find mates in the vast darkness. Bioluminescence is remarkably common in the deep sea. It has been estimated that the majority of organisms below a certain depth are capable of producing light. The deep ocean, far from being a lifeless void, is filled with a quiet, living glow, a constellation of tiny lights flickering in the darkness, each one a signal from a creature adapted to a world without sunlight. Your brain fills in blind spots in your vision every moment. There is a small area on each retina where the optic nerve exits the eye on its way to the brain. At this point, there are no photoreceptor cells, which means that no visual information is collected from this spot. It is, quite literally, a blind spot. You might expect this to appear as a dark patch or a gap in your visual field, but under normal conditions, you are completely unaware of it. The brain fills in the missing area seamlessly, using information from the surrounding region to construct a plausible continuation of the visual scene. If the area around the blind spot is a uniform color, the brain fills in that color. If there is a pattern, the brain extends it across the gap. If there is a straight line passing through the area, the brain connects the two ends. This process of perceptual filling in happens automatically and continuously, without any conscious awareness on your part. You can demonstrate the existence of your blind spot with simple visual tests, but in everyday life, it is completely invisible. This quiet act of neural construction reveals something profound about perception. What you see is not a raw photograph of reality. It is a carefully edited model, assembled by a brain that is remarkably skilled at hiding its own limitations from you. The image on your retina is upside down, but your brain flips it. The optics of the human eye work much like a simple camera. Light from the outside world passes through the cornea and the lens, which focus it onto the retina at the back of the eye. Because of the way light is refracted by the lens, the image that forms on the retina is inverted. Objects that are above your line of sight project to the lower part of the retina, and objects below project to the upper part. Left and right are also reversed. Yet you perceive the world the right way up, because the brain automatically corrects the orientation of the retinal image during processing. This inversion has been known since the work of early optical scientists, and it remains one of the most elegant demonstrations of the brain's role in constructing visual experience. In classic experiments, Volunteers who wore special goggles that flipped their visual field upside down initially found the world disorienting and almost impossible to navigate. But after several days, their brains adapted and they were able to function normally, perceiving the world as right side up even through the inverting lenses. When the goggles were removed, they experienced a brief period of disorientation again as the brain readjusted. This adaptability reveals that what you see is not determined by the image on the retina, but by the brain's interpretation of it. Perception is always a quiet act of translation. Your eyes make tiny movements even when you think you are staring still. Even when you believe you are gazing steadily at a single point, your eyes are never truly still. They are constantly making tiny, involuntary movements known as fixational eye movements. These include microsaccades, small rapid flicks that shift the eye by a fraction of a degree, as well as slower drifts and a fine tremor called physiological nystagmus. These movements are not flaws in the visual system. They are essential to its function. The photoreceptor cells in the retina are designed to respond to changes in light rather than to constant stimulation. If the image on the retina were perfectly stabilized so that it never moved relative to the photoreceptors, you would experience a phenomenon called neural adaptation, in which the image would gradually fade from awareness and eventually disappear entirely. Fixational eye movements prevent this from happening by continuously shifting the image across the retina, ensuring that the photoreceptors always have a changing stimulus to respond to. Laboratory experiments using retinal stabilization have confirmed that a perfectly still image does indeed vanish from perception within seconds. The tiny, imperceptible movements of your eyes are what keep the visual world alive and visible. It is one of the most unexpected truths about vision. The world you see exists in part because your eyes refuse to hold still, a quiet tremor in the service of sight. Peripheral vision is poor at color, but excellent at detecting motion. When you look directly at an object, the image falls on the fovea, a small pit at the center of the retina that is densely packed with cone cells, the photoreceptors responsible for color vision and fine detail. This is why the center of your visual field is sharp, colorful, and highly detailed. The periphery of the retina, by contrast, is dominated by rod cells, which are far more sensitive to light but cannot distinguish color and provide much less detail. This means that your peripheral vision is essentially colorblind and blurry. However, it excels at detecting movement. Rod cells respond very quickly to changes in light, and the neural circuits that process peripheral vision are specialized for motion detection. A flickering light or a moving object at the edge of your visual field will instantly capture your attention, even if you cannot identify its color or shape. This division of labor between the fovea and the periphery reflects the priorities of evolution. Central vision is for identifying and examining objects in detail. Peripheral vision is for monitoring the wider environment for potential threats or opportunities. It is a sentry system, a quiet guardian that watches the edges of your world while your conscious attention is focused elsewhere. Your pupils widen when you look at something you love. The size of your pupils is not controlled solely by the amount of light entering the eye. Emotional and cognitive states also influence pupil diameter. Research has consistently shown that the pupils dilate when a person looks at something they find attractive, interesting, or emotionally engaging. This response is mediated by the autonomic nervous system, the same network that controls heart rate, digestion, and the fight-or-flight response. When you see someone you love, hear a beautiful piece of music, or encounter something that captures your curiosity, the sympathetic branch of the autonomic nervous system triggers a slight relaxation of the muscles that constrict the pupil, allowing it to widen. The effect is subtle, typically only a fraction of a millimeter, but it is consistent and measurable. Psychologists have used pupillometry, the measurement of pupil diameter, as a window into emotional and cognitive processing for decades. The dilation response is involuntary and largely outside conscious control, which makes it a reliable indicator of genuine interest or arousal. Conversely, the pupils tend to constrict in response to unpleasant stimuli. This quiet physiological response connects your eyes to your emotions in a way that is both ancient and intimate. Your gaze does not merely observe the world, it reflects how you feel about it. Staring at one color makes your brain invent its opposite. If you stare at a bright patch of red for 30 seconds and then look at a white surface, you will see a phantom image in cyan, the complementary color of red. This phenomenon is known as a negative afterimage, and it reveals something fascinating about how the brain processes color. The photoreceptor cells in the retina that respond to red light become fatigued after prolonged stimulation. When you shift your gaze to a neutral surface, those tired receptors respond less strongly, while the receptors tuned to the opposing color continue to fire at their normal rate. The imbalance creates the illusion of seeing a color that is not actually present. This effect works with any color. Staring at green produces a magenta afterimage, staring at blue produces yellow, and vice versa. The phenomenon is a direct consequence of the opponent process theory of color vision, which holds that the brain processes color in opposing pairs rather than as independent channels. Negative afterimages demonstrate that color is not a property of the physical world, but a construction of the visual system. The brain creates color from the balance between competing neural signals. When that balance is disrupted, even briefly, the brain invents colors that have no external source. It is a quiet illusion, a trick of a tired retina and an ever-active brain. Light itself has no color until your brain creates it. Color does not exist in the physical world. What exists is electromagnetic radiation of different wavelengths. A photon of light with a wavelength of roughly 700 nanometers is not red. It is simply a packet of energy vibrating at a particular frequency. The experience of redness is created entirely by the brain when this particular wavelength strikes the retina and activates the long wavelength sensitive cone cells. The brain then interprets the pattern of activation across the three cone types and constructs the subjective experience of color. Outside the nervous system, there is no color, only physics. The sky is not blue, grass is not green. These are labels that the brain assigns to different wavelengths of light for the purposes of categorization and survival. Different species with different photoreceptors construct entirely different color experiences from the same physical stimulus. A flower that appears red to a human may appear ultraviolet to a bee and invisible to a creature with no color vision at all. This insight is not merely philosophical. It lies at the heart of modern neuroscience and challenges our intuitive sense that the world looks the way it truly is. Color is a quiet invention of the mind, a code the brain uses to make sense of the electromagnetic world, painted onto reality like a layer of meaning that exists only in the act of perception. A rare few people have a fourth cone type and see millions more colors. Most humans have 3 types of cone cell in their retinas, each sensitive to a different range of wavelengths. This trichromatic vision allows us to perceive roughly 1 million distinct colors, but a small number of people, almost exclusively women, may possess a 4th type of cone cell, giving them tetrachromatic vision. A true tetrachromat has 4 independent channels of color information, which in theory enables them to distinguish subtle differences in hue that are invisible to trichromatic eyes. This would dramatically expand the number of perceivable colors, potentially into the tens or even hundreds of millions. The genetic basis for tetrachromacy is understood. It arises from a variation in the genes that code for the medium and long wavelength cone pigments, which are located on the X chromosome. Because women have 2 X chromosomes, They can carry two slightly different versions of the same cone pigment, resulting in 4 distinct receptor types. However, having the genetic potential for tetrachromacy does not guarantee the perceptual experience. The brain must also develop the neural circuitry to process the additional color channel. Research in this area is ongoing, and the number of confirmed functional tetrachromats remains very small. If you are one, your visual world is painted in a palette that others can only imagine. It is a quiet gift, a spectrum richer than the already astonishing world of color. On average, men perceive light slightly differently than women. Research into visual perception has revealed consistent differences between how men and women process color and detail. On average, women tend to be slightly better at distinguishing between subtle shades of color, particularly in the green and yellow regions of the spectrum. This advantage may be related to the fact that the genes for 2 of the 3 types of cone cell are located on the X chromosome, and women with 2 copies have a slightly higher probability of carrying cone variants that enhance color discrimination. Men, on the other hand, tend to perform slightly better on tasks that involve detecting fine detail and rapid movement, particularly in the periphery of the visual field. Some researchers have suggested that these differences may reflect the different selective pressures that shaped male and female vision during the long period of human evolution, with men more engaged in hunting tasks that required motion detection and women more engaged in gathering tasks that required color discrimination. These are statistical averages, and there is enormous overlap between the 2 groups. Many men have excellent color vision, and many women have excellent motion detection. The differences are subtle and probabilistic, not absolute. They are a quiet reflection of the many small ways in which biology shapes individual experience. Goldfish can detect both infrared and ultraviolet light. The common goldfish possesses a visual system that extends well beyond the range of human sight. While human eyes are limited to wavelengths between roughly 380 and 700 nanometers, goldfish have photoreceptor cells sensitive to both shorter ultraviolet wavelengths and longer near-infrared wavelengths. This gives them a broader visual window than most mammals, allowing them to perceive light that is completely invisible to us. The ability to detect ultraviolet light is shared by many fish, birds, and insects, and is thought to be useful for a variety of ecological purposes. In the underwater environment where goldfish live, ultraviolet sensitivity may help with detecting plankton, recognizing conspecifics, and navigating in shallow water where ultraviolet light penetrates. The near-infrared sensitivity is less common and is thought to help goldfish detect objects and predators in murky or dimly lit water. These extended visual capabilities make the goldfish's perceptual world fundamentally different from our own. A pond that appears dull and featureless to a human eye may be a rich tapestry of ultraviolet and infrared patterns to a goldfish swimming through it. It is a quiet reminder that even the most familiar animals inhabit sensory worlds that are far more complex than we tend to assume. Some frogs sense light through photoreceptive molecules in their skin. While frogs have well-developed eyes, some species also possess photoreceptive molecules in their skin that allow them to detect changes in light independently of vision. These dermal photoreceptors do not form images. Instead, they provide the frog with a diffuse awareness of illumination levels, light direction, and possibly color. Research has shown that certain frog species can adjust their skin color in response to changes in ambient light, even when their eyes are covered, suggesting that the skin itself is gathering light information. The photoreceptive molecules involved are similar to the opsins found in the retina, proteins that change their shape when they absorb light and trigger a neural signal. Dermal photoreception is not unique to frogs. It has been documented in a variety of organisms, including fish, reptiles, cephalopods, and even some insects. In frogs, this ability may serve several purposes, including regulating circadian rhythms, adjusting skin pigmentation for camouflage, and detecting predatory shadows overhead. It is a distributed sensory system, spread across the entire body surface, providing a panoramic awareness of the light environment that supplements the detailed vision of the eyes. For these frogs, the skin is a quiet second eye, sensing the world in light without ever forming a single image. Mantis shrimp punch so fast they create flashes of light. The mantis shrimp is not only remarkable for its extraordinary color vision, it is also one of the most powerful strikers in the animal kingdom. Certain species, known as smashers, possess club-like appendages that they use to strike prey with devastating force. The strike is powered by a spring-loaded mechanism in the arm that stores elastic energy and releases it in a fraction of a millisecond. The acceleration is enormous, comparable to that of a bullet leaving a gun. The strike is so fast that it creates a phenomenon known as cavitation. As the club moves through the water, it creates a zone of extremely low pressure behind it. The water in this zone momentarily vaporizes, forming tiny bubbles that collapse almost instantly. When these cavitation bubbles collapse, they produce a flash of light called sonoluminescence, along with a shock wave, a burst of heat, and a loud snapping sound. The prey is struck not once but twice, first by the physical impact of the club and then by the shock wave from the collapsing bubbles. The force is sufficient to shatter the shells of crabs and snails. No other animal known to science produces light through the sheer speed and violence of a physical strike. It is a quiet miracle of biomechanics, compressed into a movement too fast for the human eye to follow. Your perception of time is a sense that can stretch or shrink. Time is not something you perceive through a dedicated organ the way you perceive light through the eyes or sound through the ears. Yet it is as real a part of your sensory experience as any of them. Your brain constructs a continuous sense of temporal flow from a combination of internal rhythms, external cues, and neural processing speed. And this constructed sense of time is remarkably flexible. When you are deeply engaged in an activity, time seems to pass quickly. When you are bored or anxious, it crawls. In moments of extreme danger, many people report that time appeared to slow down dramatically, as though the world were moving in slow motion. These subjective distortions of time are not imagined. They reflect real changes in the way the brain processes and encodes experience. During high arousal states, the brain lays down memories more densely, and this thicker memory trace is later interpreted as evidence that the event lasted longer than it actually did. Neurochemistry also plays a role. Dopamine, the neurotransmitter associated with reward and attention, can speed up or slow down the brain's internal clock depending on its concentration. Your perception of time is one of the most personal and least understood of all your senses. It is a quiet clock running inside you, shaped by emotion and attention. Fear can make time appear to slow down dramatically. Many people who have lived through accidents, falls, or near-death experiences describe the sensation that time seemed to slow to a crawl during the most frightening moments. Soldiers in combat, skydivers whose parachutes malfunctioned, and survivors of car accidents have all reported this effect. For decades, scientists debated whether fear actually speeds up the brain's processing, allowing more perceptual information to be gathered per second, or whether the effect is an artifact of memory. Ingenious experiments have shed light on this question. In one study, participants were dropped from a high platform in a controlled freefall while watching a rapidly flickering display. If fear truly sped up perception, they should have been able to read the display at a speed impossible under normal conditions. They could not. The researchers concluded that fear does not actually increase the temporal resolution of perception. Instead, it enhances the encoding of memory. During a frightening event, the amygdala triggers a flood of stress hormones that cause the brain to lay down an unusually detailed and dense memory of the experience. When this memory is later recalled, its richness is interpreted as evidence that the event lasted longer than it did. The world did not slow down. Your brain simply recorded it in higher definition. It is a quiet trick of memory, an illusion born from the intensity of fear. Your brain predicts events before you consciously notice them. The brain is not a passive receiver of sensory data. It is a prediction engine, constantly generating hypotheses about what is likely to happen next and comparing those predictions against incoming information. This predictive processing occurs on timescales faster than conscious awareness. By the time you become consciously aware of an event, your brain has already formed expectations, prepared motor responses, and compared the new data against its model of the world. This is why catching a ball feels effortless, even though the calculations involved are extraordinarily complex. Your brain has predicted the trajectory of the ball long before it arrives, and your hand is already moving to the predicted landing point. The same principle applies to all the senses. When you walk through a familiar room, your brain predicts the location of every piece of furniture, the feel of the floor beneath your feet, and the ambient sounds of the space. Only when something deviates from the prediction, a chair moved to an unexpected position, an unusual sound, a strange smell, does the brain flag it for conscious attention. This system is highly efficient. By handling expected events automatically, it frees conscious processing power for the unexpected. Your waking experience is a quiet stream of predictions, most of them confirmed and forgotten, a few of them broken and noticed. You perceive the present moment slightly in the past. What you experience as the present moment is not truly the present. It is a reconstruction assembled by the brain from sensory information that has already traveled through nerves, been processed by relay stations, and been integrated across multiple brain regions. This takes time. A visual stimulus requires roughly 50 to 100 milliseconds to travel from the retina to the visual cortex and reach conscious awareness. An auditory stimulus takes slightly less, while a touch on the toe takes longer still because of the greater distance the signal must travel. The brain compensates for these delays by stitching together information from different senses and creating a unified experience that feels seamless and immediate, even though it is not. You are always perceiving the world as it was a fraction of a second ago. For most practical purposes, this delay is irrelevant. The world changes slowly enough that a 100-millisecond lag makes no difference. But in situations that demand extremely rapid responses, such as professional sports or combat, training can sharpen the brain's predictive systems to compensate for the inherent latency of perception. The present moment, as you experience it, is a quiet construction, a polished summary delivered just slightly behind the actual flow of events. Jet lag disrupts your internal sense of time and rhythm. Your body runs on an internal clock known as the circadian rhythm, a roughly 24-hour cycle that regulates sleep, wakefulness, hormone release, body temperature, and many other physiological processes. This clock is maintained by a cluster of cells in the hypothalamus called the suprachiasmatic nucleus, which synchronizes itself to the external light-dark cycle through signals received from the eyes. When you travel rapidly across several time zones, the external light-dark cycle shifts abruptly, but your internal clock cannot adjust immediately. The result is jet lag, a temporary misalignment between your internal rhythms and the local time. The symptoms are familiar: fatigue, insomnia, difficulty concentrating, digestive disturbances, and a general sense of disorientation. Your body is still operating on the schedule of your departure city while the world around you has moved on. The circadian clock adjusts gradually, typically at a rate of roughly 1 hour per day, which means that a long-haul flight across 8 time zones may require a week or more of recovery. Jet lag is not merely an inconvenience. It reveals the depth of the body's dependence on temporal regularity. Your sense of time is not just a mental experience. It is a physiological reality embedded in every cell and organ, a quiet rhythm that prefers consistency. Humans may possess a faint magnetic sense for direction. Many animals, including birds, sea turtles, and salmon, are known to navigate using the Earth's magnetic field. For a long time, it was assumed that humans had entirely lost this ability during the course of evolution. But recent research has reopened the question. In carefully controlled experiments, human subjects placed in a chamber with a rotating magnetic field showed measurable changes in brain wave activity in response to specific magnetic field orientations. These changes were detected in the alpha wave band, a frequency associated with relaxed wakefulness, and they were consistent and reproducible. The researchers concluded that the human brain is capable of detecting geomagnetic stimuli at a subconscious level, even though we are not consciously aware of any magnetic sensation. The mechanism by which this detection occurs is not yet fully understood. One leading hypothesis involves molecules called cryptochromes, which are found in the retina and are known to be sensitive to magnetic fields in birds. Another possibility involves tiny crystals of magnetite, a naturally magnetic mineral that have been found in human brain tissue. Whether this vestigial magnetic sense has any practical influence on human behavior or navigation remains unknown. It is a quiet possibility, a faint echo of an ancient sensory ability that may not be entirely gone. Some birds and humans have molecules that detect magnetic fields. Cryptochromes are a class of light-sensitive proteins found in the retinas of many animals, including birds and humans. In migratory birds, cryptochromes are thought to play a central role in magnetoreception, the ability to detect the direction and intensity of the Earth's magnetic field. The mechanism is believed to involve a quantum effect. When light strikes a cryptochrome molecule, it produces a pair of electrons whose quantum states are influenced by the surrounding magnetic field. This subtle quantum interaction alters the chemical behavior of the molecule, which in turn generates a neural signal that the brain can interpret as directional information. In birds, this system is remarkably effective. It allows species like the European robin to navigate across thousands of kilometers during annual migrations. In humans, cryptochromes are present in the retina, but their role in magnetic perception is much less clear. The experimental evidence so far suggests that the human brain can detect changes in magnetic field strength and direction, but the response is subconscious and does not appear to guide behavior in any obvious way. Whether human cryptochromes once served a navigational function and have since become vestigial, or whether they still contribute to some subtle aspect of spatial awareness, remains a fascinating open question. It is a quiet intersection of quantum physics and animal perception. Salmon navigate home using the planet's magnetic signature. After spending years in the open ocean, Pacific salmon return to the exact freshwater stream where they were born to spawn. This navigational feat is one of the most remarkable in the animal kingdom. Research has shown that salmon use the Earth's magnetic field as a map during the oceanic phase of their journey. Each location on Earth has a unique magnetic signature, defined by the combination of magnetic field intensity and inclination. Salmon appear to be capable of reading these magnetic parameters and using them to determine their approximate latitude and longitude. This allows them to navigate across thousands of kilometers of open ocean to the general vicinity of their home river system. For the final stage of the journey, they switch to a different sense entirely. They use their extraordinary sense of smell to identify the unique chemical signature of their birth stream, following the scent upstream to the precise location of their hatching site. This dual navigation system, magnetic for the open ocean and olfactory for the final approach, is an elegant solution to the challenge of finding a tiny stream in a vast landscape. It is a quiet testament to the sophistication of animal perception, a journey guided by the invisible lines of force that encircle the planet. Pigeons sense subtle changes in air pressure as they fly. Homing pigeons have long been celebrated for their ability to find their way home over enormous distances. While much of this navigation is attributed to their use of the Earth's magnetic field, visual landmarks, and olfactory cues, research has also shown that pigeons are sensitive to subtle changes in barometric pressure. This sensitivity may allow them to detect weather fronts, altitude changes, and atmospheric gradients as they fly, providing additional navigational information that complements their other senses. The mechanism underlying this barometric sensitivity is thought to involve the paratympanic organ, a small sensory structure located near the middle ear. This organ contains hair cells similar to those found in the inner ear and is believed to respond to changes in air pressure. Some researchers have also suggested that pigeons use infrasound, very low-frequency sound waves generated by ocean waves, geological activity, and weather systems as a navigational aid. These infrasonic signals travel vast distances and could provide a consistent auditory map of the landscape. Whether pigeons rely primarily on pressure, infrasound, magnetism, smell, or some combination remains a topic of active investigation. What is clear is that the pigeon's navigational toolbox is extraordinarily rich. It is a quiet mastery of the atmosphere, a reading of the invisible currents that flow through the sky. Sharks detect electric fields through pores on their snouts. Sharks possess a sensory system that is entirely absent in humans. The ampullae of Lorenzini are a network of small, jelly-filled pores concentrated on the snout and head of the shark. Each ampulla consists of a tube connecting the surface pore to a cluster of electroreceptor cells deep beneath the skin. These cells are extraordinarily sensitive to the weak electric fields generated by the muscular contractions and nerve impulses of other animals. Every living creature in the ocean produces a faint bioelectric field, and sharks can detect these fields at remarkably low intensities. The sensitivity of the ampullae is so great that sharks can detect electric fields as weak as a few nanovolts per centimeter, a level of sensitivity that is almost unimaginably fine. This electroreception allows sharks to locate prey that is hidden from sight, buried in sand or concealed in murky water. A flatfish lying motionless on the sea floor, invisible and odorless, still produces a bioelectric signature that the shark can detect as it passes overhead. Electroreception also plays a role in navigation. Sharks may use the electric fields generated by ocean currents moving through the Earth's magnetic field to orient themselves over long distances. It is a quiet sixth sense, tuned to a dimension of the world that no human can perceive. The platypus hunts by sensing electrical signals from prey. The platypus is one of the most unusual mammals on Earth, and its bill is one of the most remarkable sensory organs in the animal kingdom. The bill is covered with thousands of electroreceptors and mechanoreceptors that allow the platypus to detect both the electrical signals and the mechanical disturbances produced by its prey. When a shrimp, insect larva, or small crustacean moves or contracts its muscles underwater, it generates a faint electric field. The platypus detects this field through electroreceptor cells arranged in rows along the bill. Simultaneously, the mechanoreceptors detect the pressure waves created by the prey's movement. The platypus hunts with its eyes, ears, and nostrils closed, relying entirely on the electroreceptive and mechanoreceptive information from its bill to find food. The brain combines the two streams of data to pinpoint the location and distance of the prey with impressive accuracy. This dual sensory system is thought to have evolved as an adaptation to hunting in the murky, freshwater environments where the platypus lives. Vision is of limited use in these conditions, so the bill provides an alternative that is both precise and reliable. The platypus's bill is a quiet masterpiece of sensory engineering, detecting the faintest electrical whispers of life hiding in the riverbed. Certain fish communicate entirely through electric pulses. Several families of freshwater fish, most notably the electric fish of Africa and South America, have evolved the ability to generate and detect weak electric fields. These weakly electric fish produce continuous electric discharges from a specialized organ in their tails, creating an electric field that surrounds their body. Electroreceptors distributed across the skin detect distortions in this field caused by nearby objects, other fish, or changes in water conductivity. This active electrosensory system allows the fish to navigate, find food, and detect predators in complete darkness or in the murky waters of tropical rivers. But the electric organ serves a second, equally important function. It is a communication device. Each species produces a characteristic discharge pattern, and individuals can modify their signals to convey information about identity, sex, social status, and reproductive readiness. Males and females of some species engage in elaborate electric courtship displays, modulating the frequency and waveform of their discharges in a dialogue that is invisible and silent to all non-electric observers. Some species can jam each other's signals, and social hierarchies are maintained through electrical interactions. It is an entire sensory and social world conducted in a medium that no human can perceive without instruments. A quiet conversation carried on pulses of electricity in the dark waters of a tropical river. Plants can sense touch and adjust their growth accordingly. Plants do not have nervous systems, but they are far from insensitive to their environment. Many species respond to physical contact through a phenomenon known as thigmomorphogenesis, a change in growth pattern triggered by touch or mechanical stimulation. Trees that grow in windy locations tend to develop shorter, stockier trunks than those in sheltered environments. Climbing plants such as vines and tendrils can detect the touch of a support structure and wrap around it within hours. The Venus flytrap closes its leaves in a fraction of a second when trigger hairs on its surface are touched by an insect. The sensitive plant, Mimosa pudica, folds its leaves rapidly when disturbed, a response thought to startle herbivores and expose thorns on the stem. These responses are mediated by changes in turgor pressure, electrical signaling between cells, and the release of calcium ions, all without any neural tissue. Research has shown that repeated touching or gentle brushing can make plants shorter and more robust, a response that farmers and gardeners have exploited for centuries without fully understanding the mechanism. Plants live in a world of constant physical interaction, responding to wind, rain, gravity, and the touch of other organisms. Their sensitivity is slow and silent compared to that of animals, but it is no less real. It is a quiet awareness woven into every leaf and stem. Trees appear to detect sound vibrations from insects. Recent research has suggested that some plants may be capable of detecting and responding to sound vibrations. In a series of experiments, scientists exposed plants to the recorded sound of caterpillars chewing on leaves. The plants that had been exposed to the chewing sounds produced significantly higher levels of defensive chemicals, such as glucosinolates, when they were subsequently attacked by real caterpillars, compared to plants that had been kept in silence or exposed to other types of vibration. The implication is that the plants were somehow detecting the vibrations of herbivory and priming their defenses in advance. The mechanism by which this might work is not yet fully understood. Plants do not have ears or anything resembling them. but they do have mechanosensitive proteins in their cell membranes that can respond to pressure changes and vibrations. It is possible that these proteins translate the mechanical energy of sound waves into biochemical signals that activate defense pathways. The research is still in its early stages, and not all findings have been replicated. But the possibility that plants can eavesdrop on the sounds of the organisms around them represents a profound expansion of our understanding of plant perception. If confirmed, it would mean that the forest is not the silent, passive place it appears. It is a quiet arena of acoustic surveillance. Even bacteria can sense light without possessing eyes. Bacteria are among the simplest organisms on Earth, yet many species are capable of detecting and responding to light. This ability, called phototaxis, allows bacteria to move toward or away from light sources depending on their needs. Photosynthetic cyanobacteria, for example, move toward light to maximize the energy they can capture. Other species move away from light to avoid harmful ultraviolet radiation. The molecular basis of bacterial light sensing involves photoreceptor proteins, molecules that change their shape when they absorb photons. These shape changes trigger signaling cascades within the cell that alter the direction of its swimming. In a remarkable study published in the early 2000s, researchers discovered that certain spherical cyanobacteria act as tiny lenses. Light entering one side of the spherical cell is focused onto the opposite side, where it activates photoreceptor molecules. By comparing the intensity of light on different parts of its body, the bacterium can determine the direction of the light source. This is functionally similar to the way an eye works, though on a microscopic scale and without any neural tissue. Bacteria have been sensing light for billions of years, long before the first true eyes evolved. Their quiet photoreception is one of the oldest sensory abilities on the planet. Your gut contains taste receptors that detect nutrients. The taste receptors on your tongue are not the only ones in your body. Scientists have discovered that the lining of the gastrointestinal tract, from the stomach to the intestines, is studded with cells that express the same types of taste receptor found on the tongue. These gut taste receptors respond to sugars, amino acids, bitter compounds, and fatty acids in the food that passes through the digestive system. Unlike the taste receptors on the tongue, however, the signals from gut receptors do not produce a conscious sensation of taste. Instead, they communicate with the brain through the vagus nerve and through the release of hormones and neurotransmitters. When the gut taste receptors detect sugars, they trigger the release of hormones that regulate blood sugar levels and stimulate appetite. When they detect bitter compounds, they may trigger nausea or slow gastric emptying, a protective response that limits the absorption of potentially harmful substances. These gut receptors play a crucial role in regulating digestion, metabolism, and satiety. They are part of the enteric nervous system, sometimes called the second brain, a vast network of neurons that lines the gut and operates largely independently of conscious awareness. Your gut is tasting your food long after your tongue has finished with it. It is a quiet internal chemist, monitoring every meal from within. The heart has its own nervous system that senses in real time. The heart is not simply a mechanical pump. It contains an intrinsic nervous system composed of roughly 40,000 neurons that form a complex network capable of sensing, processing, and integrating information independently of the brain. This cardiac nervous system, sometimes called the heart-brain, monitors the chemical and mechanical environment within and around the heart and adjusts cardiac function in real time. It detects changes in blood pressure, blood chemistry, hormone levels, and the mechanical stretch of the heart muscle, and it uses this information to regulate heart rate, contraction strength, and electrical conduction. The cardiac nervous system communicates with the brain through the vagus nerve and through hormones released into the bloodstream, but it can also make local adjustments without waiting for instructions from above. This autonomy allows the heart to respond rapidly to changing conditions, maintaining its rhythm and output even when communication with the brain is temporarily disrupted. The heart also releases hormones of its own, including atrial natriuretic peptide, which helps regulate blood pressure and fluid balance. The picture that emerges is of the heart as a sensory organ in its own right, quietly monitoring the internal environment and adjusting its behavior to maintain the stability that the body requires. It is a quiet intelligence beating within you at this very moment. Humans can smell emotions like fear and happiness in sweat. The chemical composition of human sweat changes depending on the emotional state of the person producing it. Sweat produced during moments of fear contains different volatile compounds from sweat produced during exercise or relaxation. Research has shown that when people are exposed to sweat collected from individuals who are experiencing fear, they show measurable physiological responses, including increased startle reflexes, heightened amygdala activity, and facial expressions associated with fear, even though they are not consciously aware of any change in odor. Similarly, sweat collected from people watching comedy clips has been shown to produce subtle changes in the facial expressions and mood of those who sniff it, promoting more positive emotional states. These findings suggest that humans engage in a form of unconscious chemical communication, transmitting and receiving emotional information through volatile compounds in sweat. This is not surprising from an evolutionary perspective. Many mammals use chemical signals to communicate danger, reproductive status, and social identity. Humans have not entirely lost this ability. Our olfactory system continues to process these chemical cues at a subconscious level, influencing our emotional states and social behavior without our knowledge. The air between people is never truly empty. It carries a quiet emotional signature, a chemical conversation that unfolds beneath the threshold of awareness. Dogs can detect diseases through smell before symptoms appear. The canine sense of smell is roughly 10,000 to 100,000 times more sensitive than that of a human. Dogs have approximately 300 million olfactory receptors, compared to about 6 million in the human nose, and the part of their brain devoted to analyzing scent is proportionally 40 times larger than ours. This extraordinary olfactory ability has led researchers to explore whether dogs can detect the subtle chemical changes in the body that accompany disease. The results have been remarkable. Trained dogs have been shown to detect lung cancer, breast cancer, bladder cancer, and prostate cancer by sniffing breath, urine, or skin samples, often with accuracy rates that rival or exceed conventional screening methods. They have also been trained to detect low blood sugar in diabetic patients and to identify the onset of epileptic seizures minutes before they occur. The diseases produce volatile organic compounds that alter the chemical profile of breath and body fluids. These changes are far too subtle for the human nose, but fall well within the detection range of a dog. Medical scent detection is still a developing field, and it is unlikely to replace laboratory diagnostics. But the ability of dogs to identify disease at its earliest stages offers the possibility of faster, non-invasive screening. It is a quiet partnership between species, sniffing out danger long before it becomes visible. Bees can be trained to identify explosives by scent. Honeybees have an impressive sense of smell and a remarkable capacity for learning. These two qualities have led researchers to explore whether bees can be trained to detect specific chemical odors, including the volatile compounds associated with explosives. In conditioning experiments, bees are exposed to a target scent and rewarded with sugar water. After repeated pairings, the bees learn to associate the scent with the reward. When subsequently exposed to the target scent alone, they extend their proboscis in anticipation of the reward, a measurable and reliable response. Using this technique, researchers have successfully trained bees to detect TNT, C4, and other explosive compounds at concentrations far below those detectable by electronic sensors. The advantage of using bees is that they are inexpensive, abundant, and can be trained quickly, often within just a few hours. Some research teams have developed portable devices that house trained bees and monitor their responses in real time, creating a biological detection system that could be deployed in the field. While the technology has not yet replaced electronic explosive detectors or trained dogs, it demonstrates the extraordinary sensitivity of insect olfaction. A creature whose brain is smaller than a grain of rice can achieve what some of the most sophisticated human-made sensors cannot. It is a quiet demonstration of the power packed into a tiny nervous system. Your sense of smell weakens as you age, especially for certain odors. Olfactory decline is one of the most common but least discussed consequences of aging. Studies have consistently shown that the ability to detect, identify, and discriminate between odors decreases progressively from middle age onward. By the age of 70, many people have experienced a significant reduction in olfactory sensitivity, and by 80, the decline can be substantial. The causes are multifaceted. The number of olfactory receptor neurons in the nasal cavity decreases with age. The mucous membrane that lines the nasal passages becomes thinner and drier, reducing the efficiency with which odor molecules are captured and transported to the receptors. Changes in the brain's olfactory processing centers also contribute to the decline. Certain odors are affected more than others. The ability to detect floral and fruity scents tends to decline earlier and more steeply than the ability to detect pungent or chemical odors. This has practical consequences. Reduced olfactory sensitivity can diminish the enjoyment of food, since so much of flavor depends on smell. It can also pose safety risks, as the ability to detect smoke, gas leaks, or spoiled food may be impaired. In recent years, olfactory testing has attracted attention as a potential early marker for neurodegenerative conditions, since a decline in smell is often one of the earliest symptoms. Your sense of smell is a quiet sentinel, and its gradual fading is one of the subtler changes of growing older. Some people can smell approaching rain before it arrives. The distinctive scent of rain on dry earth has a name. It is called petrichor. The word was coined in the 1960s by two Australian researchers who identified the compounds responsible for the smell. When rain falls on dry soil, it disturbs oils that have been secreted by plants and absorbed into the ground during dry periods. It also releases a compound called geosmin, produced by soil-dwelling bacteria of the genus Streptomyces. These volatile molecules are launched into the air by the impact of raindrops and carried by the wind, sometimes reaching human noses before the rain itself arrives. The human nose is extraordinarily sensitive to geosmin. It can detect concentrations as low as 5 parts per trillion, making it one of the most easily detected substances known. This sensitivity may have evolutionary origins. In arid environments, the ability to smell approaching rain would have been a valuable survival advantage, guiding early humans toward water sources. The fact that so many people describe the smell of rain as pleasant supports this interpretation. The olfactory system has a long memory, and scents associated with life-sustaining resources tend to be experienced as appealing. The smell of rain is not just a charming detail of the weather. It is a quiet sensory link to the ancient relationship between humans and the water cycle. Humans may unconsciously choose partners based on scent compatibility. Research in the field of evolutionary biology has suggested that humans are influenced by scent when selecting romantic partners, even though this influence operates largely below the threshold of conscious awareness. In a well-known series of experiments, women were asked to smell t-shirts worn by different men and to rate them for attractiveness. Consistently, women preferred the scent of men whose major histocompatibility complex, a set of immune system genes, was most different from their own. The evolutionary logic behind this preference is compelling. Offspring produced by parents with dissimilar immune genes would have broader immune protection, which would increase their chances of survival. By using scent as a proxy for genetic compatibility, the olfactory system provides a subtle but powerful guide to mate selection. The mechanism by which body odor reflects MHC type is thought to involve the breakdown products of MHC proteins, which are expressed on cell surfaces throughout the body and influence the composition of sweat and other bodily secretions. It is worth noting that this research is conducted under controlled conditions, and the role of scent in real-world romantic decisions is complicated by many other factors. Nevertheless, the findings point to a fascinating possibility that your nose may have a quiet opinion about your love life that your conscious mind never hears. Your body odor is as unique as your genetic code. No two people on Earth smell exactly the same. Your personal body odor is the product of a complex interaction between your genetic makeup, the bacterial communities living on your skin, your diet, your hormonal profile, and your overall state of health. The most significant genetic contributor to individual scent is the major histocompatibility complex, a group of genes that influence the immune system and that are among the most variable in the human genome. Because each person carries a unique combination of MHC alleles, the resulting pattern of metabolic byproducts and skin secretions is effectively unique. The bacteria that colonize your skin add another layer of individuality. Different people harbor different communities of skin bacteria, and these bacteria metabolize skin secretions into volatile compounds that contribute to the overall scent profile. Diet influences body odor as well. The consumption of garlic, onions, spices, and certain other foods can produce detectable changes in the smell of sweat. Even emotional states, illness, and medication can alter your scent. The resulting odor fingerprint is so distinctive that trained dogs can identify individuals by scent alone, and forensic researchers have explored using odor profiles for identification purposes. Your scent is as personal as your face or your voice. It is a quiet signature carried on the air, written in molecules too small to see. The smell of old books comes from slowly decaying molecules. The distinctive, slightly sweet, musty scent of old books is produced by the gradual chemical degradation of the organic compounds in paper, ink, and binding materials. Paper is made primarily from cellulose and lignin, and over time these molecules break down through oxidation and hydrolysis, releasing a range of volatile organic compounds into the air. Among the compounds that have been identified in the scent of aging paper are vanillin, which gives a faint vanilla-like sweetness, benzaldehyde, which has an almond-like quality, and various organic acids and aldehydes that contribute earthy, woody, and slightly acidic notes. The precise composition of the scent depends on the type of paper, the ink used, the binding materials, and the environmental conditions in which the book has been stored. Books made from acidic wood pulp paper deteriorate more quickly than those printed on higher quality acid-free stock. Libraries, used bookshops, and archives each have their own characteristic scent, shaped by the collective off-gassing of thousands of volumes. Researchers have even proposed using chemical analysis of paper volatiles as a non-invasive tool for assessing the preservation state of historical documents. The smell of old books is not just nostalgia. It is the quiet breath of decomposition, a library slowly releasing its chemistry into the air, molecule by molecule. Space has a distinct smell like seared metal and burnt steak. Astronauts returning from spacewalks have repeatedly described a distinctive odor clinging to their suits and equipment after exposure to the vacuum of space. The smell has been compared to seared steak, hot metal, welding fumes, and gunpowder. It is not the smell of empty space itself, since a vacuum contains no molecules to stimulate the olfactory receptors. Instead, the odor is thought to come from a variety of sources. One contributor is the outgassing of materials on the exterior of the spacecraft and spacesuit when exposed to the extreme temperatures and ultraviolet radiation of the space environment. Another is the presence of polycyclic aromatic hydrocarbons, complex carbon-based molecules that are abundant in interstellar space and that can be deposited on surfaces during spacewalks. These molecules are produced by dying stars and are among the most common organic compounds in the universe. Some researchers have also suggested that atomic oxygen, a highly reactive form of oxygen found in low Earth orbit, may contribute to the scent by reacting with the materials of the spacesuit. Whatever its precise chemical origin, the smell of space is a strange and evocative sensory experience, a quiet reminder that even the void between worlds has a signature that the human nose can detect. Some people see vivid colors when they hear music. Chromesthesia is a form of synesthesia in which sounds, particularly musical notes and chords, automatically trigger the perception of colors. For someone with chromesthesia, a C major chord might produce a vivid blue, while a D minor chord might appear as deep red. These color associations are consistent for each individual, meaning the same sound always produces the same color, but they vary between different synesthetes. Brain imaging studies have confirmed that the visual cortex of chromesthetes becomes active in response to musical stimuli, even when their eyes are closed. The colors are not imagined in the way you might picture a color in your mind's eye. They are experienced as genuinely present, often appearing as washes, streaks, or shapes in the visual field. Many musicians with chromesthesia report that the condition enriches their experience of music, adding an extra dimension that helps with composition, improvisation, and memory. Some of the most famous composers and performers in history are thought to have experienced some form of chromesthesia. The condition provides a vivid demonstration that the boundaries between the senses are not fixed. The brain is capable of creating connections between auditory and visual processing in ways that transform the experience of both. Music, for these individuals, is not just heard. It is seen, a quiet light show playing inside the mind. Others feel textures when hearing certain sounds. Auditory tactile synesthesia is a condition in which hearing a sound triggers a physical sensation of touch on the body. Someone with this form of synesthesia might feel a tingling on their fingertips when hearing a particular musical instrument, or a sensation of pressure on their cheek in response to a specific spoken word. The experiences are involuntary, consistent, and often highly specific, with particular sounds always producing the same tactile sensation in the same location on the body. Like other forms of synesthesia, auditory tactile synesthesia is thought to arise from unusually strong connections between brain regions that are normally more independent. The auditory cortex and the somatosensory cortex, which are anatomically adjacent, appear to be more tightly linked in these individuals. Studies have shown that the somatosensory cortex is genuinely activated by auditory stimuli in people with this condition, confirming that the tactile sensations are real and not simply imagined. Living with auditory tactile synesthesia can be both enriching and overwhelming. Noisy environments can produce a barrage of unwanted physical sensations, while quiet music may produce a gentle and pleasing tactile accompaniment. It is a quiet blurring of the boundary between hearing and feeling, a reminder that the senses are not as separate as they seem. A rare few can smell sounds or taste emotions. Synesthesia occurs in many forms, some of them extraordinarily unusual. While the most commonly reported types involve seeing colors in response to letters, numbers, or music, rarer variants include olfactory-auditory synesthesia, in which sounds trigger the perception of specific smells, and gustatory-emotional synesthesia, in which emotional states produce distinct tastes in the mouth. A person with olfactory-auditory synesthesia might smell lavender every time a doorbell rings, or detect the scent of burnt wood whenever they hear a particular voice. Someone with gustatory-emotional synesthesia might experience the taste of chocolate when feeling contentment, or the taste of metal when anxious. These experiences are not metaphors. They are genuine perceptual events, confirmed by the consistency of the associations and by brain imaging data showing activation of the relevant sensory cortices. The rarity of these forms makes them difficult to study, and much remains unknown about their neural basis. But they expand the known range of synesthetic experience and challenge the assumption that the senses operate in isolated channels. The brain is capable of wiring any sensory modality to any other, producing worlds of perception that most people can barely imagine. For those who live with these rare forms, the sensory world is boundlessly interconnected. It is a quiet tapestry of sound, scent, taste, and emotion, all woven together. Losing one sense can sharpen the others through brain rewiring. When a person loses one of the major senses, whether through injury, illness, or congenital absence, the brain undergoes a process of reorganization that can dramatically enhance the remaining senses. This phenomenon, known as cross-modal plasticity, occurs because the brain areas that would normally process the lost sense are not simply left idle. Instead, they are gradually recruited to process input from other senses, effectively increasing the neural resources available for the remaining modalities. Blind individuals, for example, often develop enhanced hearing, including superior ability to localize sounds in space, detect pitch differences, and understand speech in noisy environments. Brain imaging shows that their visual cortex is activated during auditory and tactile tasks. Similarly, deaf individuals often develop sharper peripheral vision and greater sensitivity to vibration. The degree of enhancement depends on several factors, including the age at which the sense was lost and the extent to which the individual actively uses and trains the remaining senses. The brain's capacity to reorganize itself in response to sensory loss is one of the most powerful demonstrations of neural plasticity. It reveals that the brain is not a fixed machine, but a dynamic organ, constantly adapting its structure to make the best use of the information available. It is a quiet resilience, a system that finds a way to thrive even when diminished. Humans can learn echolocation by interpreting sound reflections. Some blind individuals have developed the ability to navigate their environment by producing clicking sounds with their tongue or fingers and listening to the echoes that bounce back from nearby objects. This skill, known as human echolocation, is remarkably similar in principle to the echolocation used by bats and dolphins. By analyzing the timing, pitch, and loudness of the returning echoes, an experienced echolocator can determine the distance, size, shape, and even the material of obstacles in their path. Brain imaging studies of skilled human echolocators have revealed that the echoes are processed not in the auditory cortex, but in the visual cortex, the same brain region that processes sight in sighted individuals. This is a striking example of cross-modal plasticity. The visual cortex has been repurposed to construct a spatial map of the environment from auditory data. Some echolocators can ride bicycles, hike in unfamiliar terrain, and navigate complex indoor environments with surprising confidence. The skill can be taught, and training programs have been developed to help both children and adults with visual impairments learn the technique. Human echolocation demonstrates that the brain's capacity for spatial awareness is not limited to any single sense. When one channel is closed, another can step in. It is a quiet superpower born from the brain's determination to make the world navigable. People born blind often develop extraordinary spatial awareness. Individuals who are born without sight frequently develop a version of spatial awareness that relies on the integration of hearing, touch, proprioception, and echolocation rather than vision. This spatial sense can be remarkably precise. Many congenitally blind individuals can navigate complex environments, identify the location and distance of objects by sound and touch, and build detailed mental maps of spaces they have explored. The brain regions that support this enhanced spatial awareness include the visual cortex, which, in the absence of visual input, is recruited to process auditory and tactile information. This cross-modal plasticity allows blind individuals to use a broader neural network for spatial processing than sighted individuals typically employ. Studies using functional brain imaging have shown that the spatial processing abilities of congenitally blind individuals are centered in brain areas that overlap with the spatial processing networks of sighted people, suggesting that the underlying spatial architecture of the brain is modality independent. It does not require visual input to function. It can work with whatever data is available. The result is a form of spatial cognition that, while different from visual spatial awareness, is often equally effective and sometimes superior in certain respects. It is a quiet demonstration that the brain's spatial understanding is not locked to any particular sense, but is a flexible capacity that adapts to whichever channels are open. Your brain constantly merges all senses into one unified world. At any given moment, your brain is receiving a flood of information from your eyes, ears, nose, tongue, skin, muscles, joints, and internal organs. Each of these streams arrives at the brain through a different pathway and is initially processed in a different region. Yet your conscious experience is not a disjointed collection of separate sensory channels. It is a single, unified perception of the world. The process by which the brain combines these diverse streams into one coherent experience is known as multisensory integration. It is one of the most remarkable and least understood feats of neural processing. The brain uses timing, spatial coincidence, and prior experience to decide which sensory signals belong together. A voice coming from the direction of a moving mouth is bound together as one event. The smell, appearance, and taste of food are merged into a unified flavor experience. The brain achieves this integration through networks of association areas, higher-order cortical regions that receive input from multiple sensory areas and combine them. The process is so seamless that you are normally unaware it is happening. You experience a world, not a set of separate data streams. This effortless unity is the product of immense neural coordination. It is a quiet magic, performed every moment of your waking and sleeping life, holding the world together in your mind. Virtual reality can trick your brain into believing it is elsewhere. Virtual reality technology works by simultaneously delivering artificial visual, auditory, and sometimes tactile signals that together persuade the brain it is present in an environment that does not physically exist. The key to the illusion is multisensory consistency. If the visual scene shifts when you turn your head, if the sounds change as you move through the virtual space, And if the timing of all these signals is tightly coordinated, the brain accepts the artificial environment as real. This phenomenon is known as presence, the subjective experience of being inside the virtual world. Studies using brain imaging have shown that VR can activate the same brain regions that respond to real environments, including areas involved in spatial navigation, emotional processing, and threat detection. The brain's reliance on sensory consistency over absolute accuracy is what makes VR possible. Because the brain constructs reality from the best available evidence, it can be deceived whenever artificial signals are sufficiently coherent and well-timed. VR is used in medical contexts for pain management, rehabilitation, and the treatment of phobias and post-traumatic stress. It is also used in research to study perception, navigation, and social behavior under controlled conditions. The success of virtual reality is a quiet tribute to the brain's trust in its senses. If the signals agree, the brain believes. It does not question the source. When sight and sound conflict, your brain chooses the most plausible. When the brain receives conflicting information from different senses, it must decide which source to trust. In many situations, vision wins. This visual dominance is dramatically illustrated by the McGurk effect, a perceptual phenomenon in which watching a person mouth one syllable while listening to a different syllable produces the perception of a third, entirely different syllable. The brain combines the visual and auditory information and generates a percept that matches neither input alone. The ventriloquist effect is another example. When a voice is heard while a puppet's mouth moves, the brain attributes the sound to the puppet, despite the spatial mismatch. The reason for visual dominance is thought to relate to the spatial precision of vision. The visual system provides the most detailed and reliable spatial information of any sense, so the brain tends to defer to it when there is a conflict involving location or identity. However, visual dominance is not absolute. In situations where auditory or tactile information is more reliable, such as in the dark, or when timing matters more than location, the brain will prefer the non-visual sense. The brain is not blindly loyal to any single sense. It is a probabilistic system that weights each source according to its reliability in the current context. It is a quiet negotiation, happening faster than thought. You can feel motion even when standing perfectly still. Vection is the illusion of self-motion that occurs when a large visual scene moves around a stationary observer. If you have ever sat in a parked train and felt a jolt of motion when the train next to you begins to move, you have experienced vection. The visual system interprets the flowing visual scene as evidence that you are moving, and this interpretation can be so powerful that it overrides the signals from your vestibular system and proprioceptors, which correctly report that you are stationary. The result is a compelling sensation of movement in the absence of any physical displacement. Vection is central to the experience of immersive cinema, flight simulators, and virtual reality. It is also responsible for the nausea that some people experience in these environments, because the conflict between visual motion signals and vestibular stillness can trigger motion sickness. The strength of vection depends on several factors, including the size of the visual field that is filled by the moving scene, the speed of the visual motion, and the direction of the flow. Full-field visual motion in the peripheral visual field is particularly effective at producing the illusion. Vection demonstrates that your sense of movement is not a simple readout from the inner ear. It is a construction assembled from multiple sources, and vision can overrule the body. It is a quiet deception played by your own brain. Illusions occur because your brain predicts rather than records. The brain does not passively receive sensory information and present it to consciousness unfiltered. Instead, It constantly generates predictions about what is most likely to be happening in the environment and compares those predictions against incoming sensory data. When the prediction matches the data, perception is smooth and efficient. When there is a mismatch, the brain adjusts, but sometimes the adjustment produces a perceptual error, an illusion. Optical illusions, auditory illusions, and tactile illusions all exploit the brain's reliance on prediction. The Mueller-Lyer illusion, in which two lines of equal length appear different because of the orientation of arrowheads at their ends, works because the brain uses the arrowheads as depth cues and adjusts perceived length accordingly. The rubber hand illusion, in which synchronous touch on a hidden real hand and a visible rubber hand causes the brain to adopt the rubber hand as its own, works because the brain predicts that the seen and felt touch belong to the same body. These illusions are not failures of the sensory system. They are the natural consequences of a system optimized for speed and efficiency rather than perfect accuracy. By predicting rather than recording, the brain can process information faster, fill in gaps, and maintain a stable perception of the world. The price is occasional deception. It is a quiet trade-off and one that serves us well. Pain is real, but your brain can switch it off in emergencies. Pain is not a direct readout of tissue damage. It is a perception constructed by the brain, and the brain has the ability to suppress it when survival demands attention elsewhere. This phenomenon, known as stress-induced analgesia, has been documented in soldiers, accident victims, and athletes who sustain serious injuries but report feeling no pain until the crisis has passed. The mechanism involves the release of endogenous opioids, natural painkillers produced by the brain, including endorphins and enkephalins. These molecules bind to the same receptors as morphine and can dramatically reduce the transmission of pain signals in the spinal cord and brainstem. The descending pain modulation system, a network of brain regions including the periaqueductal gray matter and the rostral ventromedial medulla, can actively inhibit pain signals before they reach conscious awareness. The brain has, in effect, a built-in volume control for pain, and it turns it down when the priority is escape, defense, or protecting others. This does not mean the injury is not real. The tissue damage exists, and the pain will return once the emergency is over. But in the critical moments of a crisis, the brain makes a calculation that pain is less important than action. It suppresses a signal that would normally be overwhelming. It is a quiet act of self-preservation, a gift from a brain that knows when not to listen. The placebo effect can physically change how your senses perceive. The placebo effect is the phenomenon in which a person's perception or physiological state changes in response to an inert treatment that carries no active ingredient. When someone takes a sugar pill believing it to be a painkiller, their brain may release endogenous opioids and genuinely reduce the experience of pain. When someone drinks a non-alcoholic beverage believing it contains alcohol, their perception of their own coordination, social confidence, and even their speech patterns may change. The placebo effect is not limited to pain. It has been documented in studies of taste perception, where the same wine is rated more highly when presented in an expensive bottle, and in studies of vision, where participants told they are wearing corrective lenses report improved visual clarity even when the lenses are plain glass. Brain imaging confirms that placebo treatments can alter activity in the sensory cortices. The neural changes are real. What drives the placebo effect is expectation, the brain's prediction of what should happen based on context, belief, and prior experience. When the brain expects a change, it creates the conditions for that change to occur, adjusting neurotransmitter levels, attention, and sensory processing to match the prediction. It does not require deception. It reveals a deep truth about perception. What you believe shapes what you experience. It is a quiet power hidden inside every act of expectation. Memories are stored with sensory tags that reawaken during dreams. When you form a memory, the brain does not store it as a single file. Instead, it encodes the experience as a pattern of activity distributed across multiple sensory areas. A memory of a childhood kitchen might include the visual appearance of the room, the smell of baking, the warmth of the oven, the sound of a radio, and the taste of whatever was being prepared. Each of these sensory components is stored in the brain area responsible for processing that type of information. The visual element is stored in visual cortex, the auditory element in auditory cortex, and so on. The hippocampus acts as an index, binding these distributed fragments together so that recalling one element can trigger the others. This is why a single scent can bring back a vivid and complete memory, a phenomenon sometimes called a Proustian moment. During sleep, the brain replays and consolidates these sensory memories. In the rapid eye movement stage of sleep, the brain reactivates sensory patterns, and this reactivation is thought to contribute to the vivid, multisensory quality of dreams. You do not just see in dreams. You hear, touch, smell, and sometimes taste. The brain reassembles stored sensory fragments into new narratives. It is a quiet nightly rehearsal, a way of revisiting the world through senses that are no longer receiving external input. Taste perception changes throughout the day with your body clock. The perception of taste is not constant. It fluctuates over the course of the day in synchrony with the circadian rhythm, the internal biological clock that governs the timing of sleep, hormone release, metabolism, and many other bodily processes. Research has shown that sensitivity to sweet tastes tends to be lowest in the morning and increases as the day progresses, peaking in the late afternoon or evening. Sensitivity to salt and bitter tastes also varies across the day, though the patterns differ. These changes are driven by circadian fluctuations in the levels of hormones such as leptin, ghrelin, and insulin, which influence taste receptor sensitivity and the brain's processing of taste signals. The practical consequence is that the same food can taste noticeably different depending on when you eat it. A dessert consumed in the evening may taste sweeter than the same dessert eaten at breakfast. The circadian modulation of taste is thought to serve an adaptive function, aligning taste preferences with metabolic needs at different times of day. In the morning, when the body needs sustained energy, sensitivity to calorie-dense sweetness may be reduced to encourage a broader diet. In the evening, heightened sweet sensitivity may promote the intake of quick energy before the overnight fast. It is a quiet rhythm, ticking beneath every meal, shaping what you taste without your knowledge. Your body can sense changes in atmospheric pressure before a storm. Many people report feeling changes in the weather before they can see or hear any sign of an approaching storm. This is not mere superstition. The human body contains several systems that are sensitive to changes in atmospheric pressure. The most obvious is the inner ear, which relies on pressure differentials to function properly and can be affected by rapid barometric changes. Sinus cavities and joint capsules are also sensitive to external pressure. When atmospheric pressure drops, as it commonly does ahead of a storm, the tissues inside joints can expand slightly, potentially triggering discomfort or stiffness, particularly in people with arthritis or previous injuries. Changes in barometric pressure can also affect blood pressure, circulation, and the behavior of dissolved gases in the blood. Some researchers have found correlations between falling barometric pressure and the onset of migraines, suggesting that the brain itself may be sensitive to pressure changes. The trigeminal nerve, which innervates much of the face and head, is a candidate pathway for this sensitivity. While the mechanisms are still being studied and the evidence is not conclusive in all cases, there is growing recognition that the body possesses a form of atmospheric sensing that lies outside the traditional five senses. It is a quiet barometer embedded in the tissues, offering a wordless forecast of what the sky is about to do. Many people sense when someone is staring at them. The feeling that someone is watching you, even when you cannot see them, is one of the most commonly reported and most debated sensory phenomena. Many people are convinced they can detect the gaze of another person without any visual or auditory cue. Scientific investigation of this phenomenon has produced mixed results. Some studies have found that people can detect being stared at with above-chance accuracy under certain controlled conditions, although the effect sizes are typically small. The mechanisms that might explain this ability are unclear. One possibility is that peripheral vision, which is highly sensitive to movement and direction of gaze, provides subtle cues that the conscious mind does not register, but the brain processes unconsciously. Another possibility involves sensitivity to very slight environmental cues, such as the sound of someone shifting their body or the subtle change in social atmosphere when someone focuses attention on you. Some researchers have explored more exotic explanations, including sensitivity to electromagnetic fields, though these hypotheses remain unsupported by mainstream science. Skeptics argue that the experience is better explained by confirmation bias, the tendency to remember the times when you turned around and found someone looking at you while forgetting the many times you turned and no one was there. Whatever the final explanation, the phenomenon highlights the brain's vigilance for social attention. It is a quiet sentinel, always scanning for the gaze of others. Some individuals feel whether changes in their joints. People with arthritis, old fractures, and other musculoskeletal conditions have long reported that their symptoms worsen before changes in the weather, particularly before rain or storms. While this claim was once dismissed as folklore, a growing body of research suggests there may be a physiological basis for it. One leading hypothesis is that falling barometric pressure, which commonly precedes wet weather, allows the tissues surrounding joints to expand slightly. In joints already affected by inflammation or damage, this expansion can press on nerve endings and increase the sensation of pain or stiffness. Another hypothesis involves changes in humidity and temperature, which may affect the viscosity of synovial fluid inside the joint capsule, making movement less smooth. Some studies have found statistically significant correlations between barometric pressure drops and increases in joint pain reporting, though the relationships are often small and vary between individuals. The sensitivity appears to be greater in people with pre-existing joint conditions, suggesting that damaged or inflamed tissues are more responsive to external pressure changes than healthy ones. It is worth noting that not everyone with joint conditions experiences weather-related pain, and psychological factors such as attention and expectation may also play a role. Nevertheless, the phenomenon points to the body's quiet ability to register atmospheric changes through pathways that lie outside the traditional senses. It is a meteorological intuition, written in the aching language of the joints. Background sound can change how food tastes. The environment in which you eat has a measurable effect on how your food tastes, and one of the most powerful environmental influences is background sound. Research has shown that loud noise suppresses the perception of sweetness and saltiness while enhancing the perception of umami. This is one of the reasons why airline food is often criticized for tasting bland. The cabin noise on a commercial aircraft can reach levels that significantly dampen the taste of sweet and salty compounds, while the umami component of tomato juice remains intact, which may explain why tomato juice is disproportionately popular among air travelers. The mechanism behind this effect appears to involve multisensory interaction at the level of the brain. The chorda tympani nerve, which carries taste information from the front of the tongue, passes through the middle ear, an anatomical relationship that may allow loud sounds to physically interfere with taste signal transmission. In addition, attentional resources are diverted by loud noise, reducing the brain's capacity to process subtle taste differences. Music, as distinct from noise, can also influence taste perception. High-pitched music tends to enhance the perception of sweetness, while low-pitched music enhances bitterness. Restaurants, food designers, and the airline industry have all begun to use these findings to shape the eating experience. It is a quiet crossover between the ears and the tongue, a reminder that flavor is not just on the plate. Color can influence how something feels or smells. The color of an object or environment can change the way you perceive its smell, taste, texture, and even its temperature. Studies have demonstrated that identical fragrances presented in different colored solutions are rated differently by participants. A cherry-scented liquid-colored orange is frequently identified as smelling of oranges, while the same liquid-colored green is identified as lime. Red-colored rooms are perceived as warmer than blue-colored rooms at the same temperature, and food presented on red plates is often rated as more intense in flavor than the same food on white plates. These cross-modal influences occur because the brain uses color as a contextual cue to generate expectations about what the other senses should detect. When those expectations are strong enough, they can bias or override the actual sensory input. The visual system, because it typically provides the most spatially detailed and reliable information, tends to dominate in situations of ambiguity. Manufacturers, marketers, and designers have long exploited this effect. The color of packaging, lighting, and even the color of the container can shift consumer perception of products without changing the product itself. The phenomenon reveals how deeply interconnected the senses truly are. No sense operates in isolation. What you see shapes what you smell, what you taste, and what you feel. It is a quiet collaboration between channels that were never as separate as they seemed. The aroma of coffee can improve alertness without drinking it. Research has demonstrated that simply smelling coffee can produce measurable improvements in cognitive performance, particularly in tasks that require sustained attention and analytical thinking. In one study, participants exposed to the aroma of coffee performed significantly better on a standardized reasoning test than those in an unscented room, even though no coffee was consumed. The mechanism is likely a combination of classical conditioning and direct neurochemical effects. For habitual coffee drinkers, the smell of coffee has become strongly associated with the experience of caffeine-induced alertness through years of paired exposure. The brain, anticipating the arrival of caffeine, may begin to mobilize attentional resources in response to the scent alone, a placebo-like conditioned response. There is also some evidence that certain volatile compounds in coffee aroma may have mild psychoactive effects when inhaled. The olfactory system has a direct connection to the limbic system and the frontal cortex, brain regions involved in arousal, mood, and executive function. This direct neural pathway means that odors can influence brain state rapidly and without requiring conscious processing. The finding has implications beyond coffee. It suggests that the sensory environment, including ambient scents, can shape cognitive function in ways that are subtle but real. The smell of coffee is not just a promise of a drink. It is, in itself, a quiet nudge toward wakefulness. Certain sounds can make you shiver through a response called frisson. Frisson is the involuntary physical sensation of chills, tingling, or shivers that runs along the skin, typically down the spine and across the scalp, in response to a powerful emotional stimulus. It is most commonly triggered by music, particularly by unexpected harmonic shifts, sudden crescendos, the entry of a solo voice, or a moment of sublime beauty in a composition. Not everyone experiences frisson to the same degree. Research suggests that individuals who score highly on the personality trait of openness to experience are more susceptible. Brain imaging studies have shown that frisson activates the reward centers of the brain, including the nucleus accumbens and the ventral tegmental area, the same circuits involved in the pleasure of food, social bonding, and other basic rewards. Dopamine release has been measured during musical frisson, confirming that the experience is neurochemically real and not merely subjective. The physical component of frisson, the goosebumps and shivers, is mediated by the sympathetic nervous system and involves the contraction of tiny muscles at the base of hair follicles. This is the same piloerection response that occurs during cold exposure or fear, suggesting that frisson may be an evolutionary echo of an older arousal mechanism repurposed for aesthetic experience. It is a quiet storm of pleasure, passing through the body in response to nothing more than a well-placed note. ASMR triggers real physical tingles through soft sounds. Autonomous sensory meridian response, commonly known as ASMR, is a tingling sensation that typically begins on the scalp and spreads down the back of the neck and along the spine in response to specific auditory or visual stimuli. Common triggers include whispering, soft tapping, the crinkling of paper, gentle brushing sounds, and slow, deliberate hand movements. The sensation is accompanied by a deep feeling of relaxation and well-being, and millions of people seek out ASMR content specifically to help with sleep, stress relief, and anxiety reduction. Scientific investigation of ASMR is still in its early stages, but brain imaging studies have revealed that ASMR activates regions associated with reward, social bonding, and emotional regulation, including the medial prefrontal cortex and the nucleus accumbens. Physiological measurements during ASMR show reduced heart rate and increased skin conductance, a pattern consistent with a calm but attentive emotional state. Not everyone experiences ASMR. Sensitivity to the triggers appears to vary across individuals and may be related to personality traits such as openness and neuroticism. The precise neural mechanisms remain under investigation, but the phenomenon is increasingly recognized as a genuine perceptual experience rather than a purely subjective fantasy. ASMR reveals that the brain can derive profound physical comfort from the gentlest of sensory inputs. It is a quiet gift, a reminder that sometimes the softest sound carries the greatest power. Even silence has a sound made by your own body. If you were to sit in an anechoic chamber, a room specifically designed to absorb all external sound, you would discover that silence itself is not truly silent. Within minutes, you would begin to hear sounds coming from your own body. The high-pitched ringing or hissing you might notice is likely the spontaneous activity of the auditory system itself, known as tinnitus, which is normally masked by ambient environmental sounds. Beyond this, you would begin to hear the rhythmic thump of your heartbeat, the rush of blood through the vessels near your ears, the gurgling of your digestive system, and even the creaking of your joints as you shift your position. In extremely quiet environments, some people report hearing the faint hum of their own nervous system. The experience of an anechoic chamber can be disorienting. Humans are adapted to a world that is never truly quiet, and the absence of external sound can make the brain hyperaware of internal signals it normally suppresses. Some people find the experience distressing, while others find it strangely meditative. The body is a machine that never stops generating sound. Blood pumps, lungs inflate, muscles contract, nerves fire. True silence, in the absolute sense, is an impossibility for a living organism. It is a quiet irony that the closest you can come to silence is hearing the sound of your own existence. Sleep alters your senses, by filtering sounds to protect rest. During sleep, the brain does not simply shut off its sensory systems. Instead, it actively filters incoming signals, allowing some to pass through to awareness while suppressing others. The thalamus, a relay station deep in the brain, plays a central role in this gating process. During the deeper stages of non-rapid eye movement sleep, the thalamus reduces the flow of sensory information to the cortex, making it harder for external stimuli to reach conscious awareness. However, the suppression is selective. Research has shown that the sleeping brain continues to process meaningful sounds. A person's own name, spoken quietly, is more likely to trigger arousal than an unfamiliar name. A parent sleeping through urban noise may wake instantly at the quiet cry of their child. This selectivity suggests that the brain maintains a form of sentinel processing during sleep, evaluating incoming sounds for significance without fully waking. The auditory cortex has been shown to respond to sounds during sleep, and the brain can even perform basic linguistic processing, categorizing heard words as real or nonsensical. The balance between sensory suppression and selective attention during sleep is essential for rest. Too much suppression, and you would fail to respond to danger. Too little, and you would wake at every small sound. It is a quiet calibration, performed anew each night, that allows you to sleep peacefully in a world that never truly falls silent. Your perception of reality is a symphony your senses compose together. Everything you have ever experienced, every sight, sound, smell, taste, and touch, has been constructed inside your skull by a brain that never directly contacts the outside world. The brain sits in complete darkness and silence within the bone vault of the cranium, receiving only electrical impulses transmitted along nerve fibers from the sensory organs. From these signals, it builds the entire world you experience. The colors of a sunset, the warmth of a hand, the flavor of a favorite meal, the melody of a song, none of these exist in the form you perceive them outside of the brain. Light has no color until the visual cortex creates it. Air pressure waves are not music until the auditory cortex translates them. Molecules have no scent until the olfactory system assigns one. The brain is the composer, and the senses are the instruments. Each one plays its own part, and the brain weaves them together into a seamless, unified experience that feels as solid and real as anything could ever be. Yet it is, in the most fundamental sense, a creation, a model of the world, not the world itself. This model is astonishingly accurate most of the time, but it can also be fooled, altered, and expanded. The illusions, the synesthesias, the phantom sensations, the placebo effects you have encountered tonight all reveal the creative power of the brain. Perception is not passive. It is a symphony, and your brain is conducting it every moment of your life. And now, the quiet wonders of the senses begin to gently fade into the stillness of the night. You have wandered through over 100 peaceful truths about the way your body speaks with the universe. From the colors your brain invents to the sounds your ears never stop hearing. From the scents that unlock decades of memory to the touch that comforts before words arrive. From creatures that taste with their feet to the strange symphony of synesthesia. You have explored how light becomes sight and vibration becomes sound, how your brain fills in what is missing and predicts what comes next, how pain can vanish in moments of crisis, and how silence still hums with the rhythm of your own body. You have learned that perception is not a window, but a creation, a continuous act of translation between the raw physics of the world and the lived experience of being alive. You are connected to this story more deeply than you may realize. Right now, as you listen, your senses continue their quiet work. Your skin feels the air around you. Your ears catch the softest sounds. Your heart beats in its own gentle rhythm. These mechanisms have been refined across billions of years of evolution, passed down through every ancestor who ever lived. If your eyes feel heavy now, let them close. Let the rhythm of your breath become steady, like the slow pulse of perception beneath awareness, like the gentle hum of a nervous system that never stops caring for you. Tonight, as you rest, your senses will continue their ancient work. They will guard your sleep, filter the sounds around you, weave fragments of touch and light into dreams, prepare you gently for tomorrow. You are a warm presence within a world of endless sensation, peacefully resting among the wonders of perception. Sleep well, sweet dreams, and may you carry with you the quiet knowledge that you are never truly alone in the darkness, for your senses are always reaching outward, translating the universe into something you can feel, hear, see, and understand. That every moment of awareness is a small miracle, a conversation between your body and the cosmos. Thank you for joining me on this journey through the remarkable world of the senses. If you enjoyed this video, please subscribe to Drowsy Discovery for more soothing explorations of the wonders hidden within your own body. There are more wonders waiting, more secrets hidden in the depths of perception and the quiet corners of the mind, more gentle truths to drift through on quiet evenings. Rest well, and may the quiet symphony of your senses keep you company through the night. This has been Drowsy Discovery. Good night.